8, 9, 10, 12, 13, 14.
Sí, sí.
8, 9.
welcome, everyone. Can you hear me? It's okay. So, welcome, distinguished colleagues, dear guests. It is a great honor and a true pleasure to welcome you to the 18th edition of the Granada Seminar on Statistical and Computational Physics, organized by the Carlos First Institute for Theoretical and Computational Physics of the University of Granada. For more than 3 decades now, the Granada Seminar serves as a remarkable forum for scientific exchange, bringing together researchers from different disciplines and generations. And this conference is unique and is interdisciplinary, and its interdisciplinary spirit and its strong educational purpose with a special emphasis on supporting young scientists. Offering early career researchers the opportunity to present their results and engage in dialogue with world-renowned experts is one of the defining values of this year. This year, from September 15 to 18, we gathered here at the School of Civil Engineering on the Fontenova campus of the University of Granada to focus on the main topic, new tools and methods for complex systems research from molecules to living and intelligent systems. This topic reflects the growing importance of complexity as a fundamental concept across many areas of knowledge. As an example, recent Nobel Prize in Physics and Chemistry underscored the richness and productivity of this field, which span from molecular biology to ecosystem, from the study of the brain to artificial intelligence, and from individual to modern societies. Over the coming days, we will have the privilege to listen to distinguished plenary speakers who will guide us through a wide variety of perspectives on complexity, including microbial networks, biomolecular simulations, machine learning applied to biology and quantum complex systems, the principles underlying the origin and evolution of life, and recent theoretical development in information theory for studying complexity. We are confident that their contribution will inspire stimulating discussions, new insight, and fruitful collaboration. At the same time, I would like to extend a very warm welcome to the city of Granada. Beyond the seminar itself, we hope you take the opportunity to enjoy its unique cultural and historical heritage. From the majesty of the Alhambra and the charm of the Albaicín to the vibrant life of the city center, Granada offers an extraordinary setting where history and modern life meet. And of course, you will also discover its gastronomy, its tradition, the hospitality of its people. We sincerely hope that this environment will enrich your experience both scientifically and personally. On behalf of the organizing committee, I would like to express our deep gratitude for your presence. We hope that this seminar will be, once again, not only a place for scientific excellence, but also a space for exchange, inspiration, and community. So thanks for your presence. I will pass now to the director of the Institute Carlos Primero of the Professional Physics.
Thank you, Joaquin. Thank you, everybody, for being here. So it's a big pleasure to welcome you to the 18th edition of the Granada Seminar, director of the Institute, who is hosting this event. And for me, it's a very it's a touching moment because it's my 18th time attending this seminar. I think I'm the only one, and I say positively that I'm the only person who has attended to the previous 17 editions, and to this 18th. We have Pedro Garrido, who came to 17, but this time is in the States, working with Joel Lebovitz, so he will not be here. So now I'm taking advantage. Welcome. I'm taking advantage. I'm now the only one attending the 18th seminars. So the first edition I came was in 1990. I was a student. I had just finished my undergraduate students. So it was the very first conference I attended. I was just helping around and things. I was starting a thesis on chaos in astrophysics with Eduardo Atenar, who is here. And that was my first contact with science and everything. Then in 92, second edition, was my first talk in an international conference in the Granada Seminar. That was in Almuñécar by the coast now. A lot of fun in those times. 94, I was just finishing my PhD, so it was my first talk as a postdoc before just going to the States. 96 was my fourth edition. I came from the States with my advisor there, Jeff Greenstein. People here know him. I invited him, we invited him, and he came. It was fun. And from there, I keep on coming every year. When I was abroad, coming to Granada, and when I was local, helping to organize. And now that I'm becoming one of the oldest, I'm the organizer and the director of the institute. So it's really an honor to be here presenting this. I hope it's not very soon in the future that they organize the Granada Seminar in memoriam of Miguel Munoz, who attended 20 plus edition or something. I hope that happens in very, very far away in the future. So I think that's everything I had to say. Well, I have to thank Joaquin Torres, who was the real organizer of this edition. He took care of doing everything, which is not very easy. I won't enter the details, but it's technically complicated to organize these things. And what else should I say? Nothing. Welcome. I hope you enjoy Granada and the Granada Seminar, and that you have fun, not just presenting your results in the talk, but talking in the coffee breaks and interacting with people. That's what complexity comes about, interactions and emergence of things when we all come together and discuss. So thank you, Joaquin.
So I will pass now to the director of the Granada University.
Thank you, Joaquin. Sorry if I made the delay. Normally, I attended this opening ceremony in the time, but when you say me 50 minutes to 9, I suppose that the opening ceremony is enough. No problem. Only to short comment. Initially, I want to congratulate the organization, especially to our Faculty of Science, because this is the 18th edition of this seminar, very important seminar about statistics and physics. In representation of our rector, I want to express my gratitude to the organization, to the Faculty of Science to do that in the last 18 years, to express and to say you welcome to Granada. Now, we had the initial teaching activity in Granada. Today is the first day.
Sorry. 18th edition, 35 years. 35 years since the first one.
Oh, okay.
So it's getting
Too much. Okay. But I say you that welcome to Granada. Today we start the activity, teaching activity in Granada. You are sharing in Granada that we have very young city with a lot of younger people. Granada University is a big university with more than 55,000 students. And today that ICU is the beginning of this course. Additionally, now we have the opportunity to enjoy in Granada with the good weather. Even now it's a little bit hot, similar to the summer. We had a small summer weather during this time. The conference is 3 days, 4 days. Oh, you have time not only to work here in the night and in the afternoon. It's a good moment to visit the different part of Granada for many people that you can hear outside to Spain. I am from Europe, USA, and that's from Asia, even. In any case, welcome to Granada. I express my gratitude again to the organization. I wish you all the best in this important meeting for us and for you, not only to interchange ideas about the topic. I hope that you develop very important collaboration projects from the future. Good job for all. I hope that you organize the next year, the same conference to continue. It's the historical conference to the time. I suppose that this is a special conference for Faculty of Science. I remember that you said me, but especially for all members of the community that attending this meeting. No more. Enjoy.
So welcome again to this meeting, and let us officially open the 18th edition of the Granada Seminar.
Good morning, everyone. So my name is Paulo Hurtado. I'm also from the local organizing committee, and I'll be your chairman in this first session. So let's start with the science. So we start this morning. We have the pleasure to have here Professor Herbert Levin from the Center for Theoretical Biological Physics at Northeastern University in the US, who is going to talk about how physics can help make sense of cancer immune system dynamics. So please, Professor Herbert, come here. Thank you.
Okay, good morning. So I just realized that the last time I was in Granada was for the second Granada Seminar in 1992. And it was an interesting time because many of us were working on non-equilibrium physics, non-equilibrium physics applied to pattern formation in particular. And it was a time, for me at least, of transition where we had learned significant amounts of new ideas in that field that we felt that it was possible to begin to apply them to biological systems. And so starting from almost exactly that time, I switched my interest from sort of pure statistical non-equilibrium physics to trying to apply those ideas in biology. And I'll give you today one example of the work we've done over the last couple of years applying these ideas to the immune system. Now, if you look at the interaction between the immune system and cancer, which is what I'm going to talk about, what you discover is that it's almost a canonically multi-scale problem to think about. What I'll talk about mostly today is the interaction at the molecular scale, where in order to decide that if you have a cancer, it's something that should be attacked by the immune system, the initial effort is done at the molecular level. You need to recognize that a cell is something that does not belong to your normal body and therefore should be targeted. And that requires recognition at the molecular level, and that's what I'm really going to focus on today. But that's only the beginning of a very complex story. There's also cellular level interactions, which are indicated in the picture down here on the bottom left, that in fact, those of you who keep track of more the medical end of this topic know that recent years, there's been great progress in actually developing drugs that enable the immune system to more effectively kill cancer. It goes by the name of immunotherapy. And the breakthroughs really involved understanding cellular level signaling between it assumes that the cancer can be recognized and that you just need to give it some boost of effectiveness, and that led to development of drugs that now are effectively used for many patients. But it doesn't only end at the cellular level. There's also interactions that there you go, at the tissue level, that one of the reasons why immunotherapy might fail is that the immune cells might be physically excluded from the tumor. You see in this picture, yellow dots there are immune cells, blue is tumor regions, and you see very clearly that the yellow dots have aggregated to the tumor, they know that there's something wrong there, but they cannot access the interior of the tumor to actually contact the cells and kill them. So that's a tissue level problem. And then there's even an ecology level problem of, we think of, immune cells as one type of cell, but in fact, there are many, many types of immune cells. They all interact with each other in complex ways, and that leads to a more general problem. You can think of it as the tumor ecology. Anyway, so why is there so many layers in this immune system? Why not just recognize things at the molecular level and be done with it? And the answer is the immune system is a very complex story of how our body needs to balance different possibilities. For the immune system, it's really very dangerous in a sense, because the immune system can kill our normal cells. And in fact, there are many autoimmune diseases. Maybe the most common one is type 1 diabetes, where the immune cells go and kill all the cells in our body that produce insulin, giving diabetes, which until half a century or a little more than that by now, used to be a uniformly deadly disease. And so there needs to be a continual set of checks and balances that allow the immune system to carefully decide and make intelligent decisions based on measuring its environment, measuring other cell properties, whether or not this is something worth attacking. And if that goes wrong, it can lead to very dangerous consequences. The basic purpose of the immune system is, first of all, just keeping our tissues sort of, functioning in a sort of maintenance way, but also attacking things like very fast-growing foreign invaders, viruses, bacteria, etc. It was not really designed, in a sense, to attack cancer, and there are evolutionary arguments for that. Usually the evolutionary argument goes like, cancer is most prevalent for old people, like me, and therefore there's less evolutionary pressure to develop effective treatments to attack cancer, because after all, from the point of view of procreating future generations of the human species, I'm sort of past that stage of my life, and so it has less evolutionary pressure, whereas of course, in the pre-modern world, being able to effectively target bacterial and viral diseases is really something which is a challenge, which will kill you in early childhood, etc., etc. Okay. Now, we do work on the other aspects of this problem. We don't only work at the molecular level, and so anybody interested, I'll be here for most of the meeting. I'm happy to talk with anybody about these things at the tissue level. We've worked on this issue of this physical barrier of immune cells entering into the tumor, and we have a number of works on that over the last few years. And we also work on what I think of as this ecology problem, trying to understand how different types of cells interact in this environment, and we have some work on that. But today I'm not going to talk about that. I'm going to talk about the level of molecular detection. Now, how does the immune system decide that a tumor cell, cancer cell, is something that's worth attacking? So the answer is that tumor cells look like our cells, but there are some significant differences. One difference, the one that is which is usually the most, let's say, actionable difference, is called tumor neoantigens. Tumors have a wide variety of mutations in their genome, and that means that when they make proteins, many of the proteins are not exactly the same amino acid sequence as our proteins. And if you have a protein that has a different amino acid sequence, you should be able to recognize it as something which is not a native cell, not something which is natural to us. So that's what we're going to talk about, but it's a little bit, and this is sort of how we got interested in this problem in the first place, because it sounds a little surprising that this can work, because mutation. I mean, the genome is mutated in a tumor, but if you look at a typical protein, either it has no mutations, certainly in many proteins, or at most it would have, let's say, one mutation. So at most, the sequence of amino acids in that protein would be one amino acid away from a native sequence. And yet, we know for sure, because we see experimentally, that that one amino acid distance away from a native sequence is enough to provide information to the immune system to attack those tumor cells. And here is just one, example out of many papers from the biology literature, which just looked specifically for immune cells. The immune cells that do the attacking are these orange balls here. These are T cells, for those of you who are into the immune system, members of the adaptive immune system. And directly they could detect that, these T cells were being, activated and attacking a cell based on one amino acid change in a protein sequence. Okay. Now, the other evidence we have that mutational changes and these what are called neoantigens, antigen just means something that excites the immune system, and neoantigen just means it's sort of not quite the same as a viral sequence or bacterial sequence, but it still creates an antigenic response. So the other piece of evidence for this is that if you look at the medical literature, and this is just some paper surveyed in a few years ago, different tumors have different numbers of mutations. Now, the reason for that is sort of obvious. If you look on the far right, the one that has the highest level of mutations is melanoma. Melanoma is tumors of your skin, and the reason it has the highest number of mutations is that your skin is constantly attacked by UV radiation from being out in the sunshine, which I'm sure you have lots of here in Spain, and even in Boston, we have lots of sunshine in the summer. And that causes a large number of damage to the DNA, which then eventually creates mutations in your genome. And you can go down the line and you can compare which tumors have large numbers of mutations, which numbers have small. Another one that has a large number of mutations is various types of lung cancer, particularly the ones that are due to smoking. Because again, the problem with smoking is that the smoke, the byproducts of the cigarette smoke actually are mutagenic. They create mutations in your DNA, and so therefore, somewhere on the right-hand side of this figure is various types of lung cancer. On the left-hand side are ones that are less mutagenic, that just don't have that many mutations. So if you check if you go and listen to the TV ads, or you look at any medical literature and you see which tumors now are being effectively treated by immunotherapy, i.e. that we've been able to convince the immune system to really attack the tumor, those are the ones on the right-hand side here. The ones that have larger number of mutations provide more targets for the immune system. It's easier for them to recognize. The ones on the left typically have not been successful in applying immunotherapy. So it's a mixed story. It's great progress. Immunotherapy was given the Nobel Prize in physiology some number of years ago for discovering how to do this on the cases to the far right, but we still don't know how to do this in general. Okay. So let's go down and ask, how does this work on the molecular level? What is the recognition? So here is a picture of what this, of what the recognition looks like. So here's a T cell, member of the adaptive immune system. It has a receptor on its surface. The receptor, if you want to draw a picture of the protein, looks like sort of this thing. And then it interrogates, it means it binds to or tries to bind to other cells in your body. And those other cells in your body can be tumor cells, they can be normal cells, whatever. And these cells have a different protein on its surface, which is this thing over here. And this thing is called MHC complex. The names don't matter, but the MHC complex is a display system. It takes pieces of proteins in your body, chops them up, takes segments of those proteins, so peptide sequences. For the most important type of T cell in the cancer context, it's only 10 amino acid long sequence. So it's a peptide cut out of a protein. It puts that in some pocket. The T cell comes along, tries to bind to it, and sees if it succeeds, then it actually knows it should kill the cell. If it doesn't succeed, it says, okay, I'm just going to go on and interrogate the next cell. So it's a molecular recognition event. There's a receptor in the T cell, and then there's something it's trying to interrogate and see if it binds to it. And if it does, then it transmits a signal to the cell saying, go kill that other cell. Now, the T cells in our body are somewhat the adaptive immune system is somewhat unique because most of our cells, have unique proteins. They don't have variable proteins, but the T cells and the B cells, the parts of our immune system, have a very specialized system where they can generate a huge variety of different genomic structure, genomic patterns for these T cells. The details of how that works has been studied in great detail by work by Alexandra Walchak and collaborators from the École Normale in Paris. And in general, you can generate 10 to the 11th different T cell sequences that are then put on T cells in your body. They come out being made, each with its own independent sequence. And actually, in our blood system, if you go and check, there are sort of 10 to the 7th different T cell sequences, cells with different T cell sequences wandering around your blood system at any given time. Now, it's not that you have 10 to the 7th cells. You have many more cells than that, but they're 10 to the 7th different sequences. And if you have a few cells of those, that probably means those cells have never detected anything that they're worth attacking. If you have many cells in that particular clone, that particular T cell receptor structure, it means that you had an infection at some point. That infection was recognized by that T cell. The T cell was then given instruction to reproduce, make a lot of itself, and enable to attack the infection. And even when the infection is cleared, you still maintain a large reservoir of those cells waiting for the next infection to come by. That's why, of course, when you get a second infection of the same kind, your adaptive immune system is good at rapidly responding to that. Whereas the first time, it takes a few days and sometimes may not even succeed. Okay, so the problem we can now state that we're interested in was, given a peptide, given a specific peptide that's displayed by the cell, will it be detected by a specific T cell sequence? Okay. Now, the problem with that is, how does the cell know to ignore the sequences that are on our normal cells? Right? The logic is T cells are generated randomly by some stochastic process that's pretty well understood. They go and interrogate cells. If they happen to bind to this peptide sequence, they kill the cell. What's to stop them from happening to bind to a peptide sequence that's supposed to be on our cell, a normal protein piece in our proteome? And the answer is that the T cells go to some type of training school. But they go through a process. They're called T cells because this process occurs in the part of our body called the thymus. And in the thymus, what happens is a T cell precursor comes in. It goes through the thymus. It's exposed to many, many different peptide fragments from our body. The most common proteins in our body are broken up, put on cells in the thymus, and in the thymus, if it recognizes those cells in the thymus, the T cell is constructed to commit suicide. So it goes to school saying, if you pass the exam, that means you should kill yourself. Not like hopefully the normal schools that we all go to. So now you can understand that, of course, this process takes time. There are many different peptides that are part of our protein system. The estimates that, of how many tests it has to pass is sort of 10 to the 4th, 10 to the 5th or so. So it's probably not going to be perfect. We'll see how that works as we go on. But in any case, there's a specific process called negative selection, negative training, which attempts to delete any T cells that just by accident happen to recognize our own cells. So now, when these cells are released into our blood system, most of the time, not always, but most of the time, they will not react to any of the peptide fragments of any of our common proteins, but they will react to something which is new, they've never seen before in this training. That's the logic of the system. So the questions that you can ask if you look at this process and you think about the molecular recognition is, can we, first of all, the holy grail is, can you make an absolute prediction of when binding will occur? Can somebody give you a peptide sequence and a T cell sequence, and you can decide, via computation, via whatever methods you have, if it's going to bind or not? That's the problem everybody would like to solve, in a statistically accurate way, because then we can predict everything we want to know about this molecular recognition. That's hard, and you'll see that the community is not yet close to that. We want to understand, given the first part, we want to understand how the immune system can recognize tumor neoantigens. In other words, if you're trained to ignore a series of peptides, why does that allow you to not ignore, to bind strongly to ones that are only one amino acid away? How do you obtain that level of sensitivity that a one amino acid change out of 10 is going to actually change you from not binding to binding? Of course, you can ask if this prediction is clinically useful. The answer is definitely yes, by the way. I don't work on the clinical side, but it definitely is yes, because if you can design T cell sequences that recognize peptides from, let's say, COVID-19 or from other pathogens, then you can actually go these days and give vaccinations or other procedures to actually enhance, or you can actually even inject T cells with a specific recognition receptor into your body, and you can actually then do a better job fighting that disease. And of course, one of the things you can ask yourself in the modern world is there some way that modern progress in machine learning can help this? It sounds somewhat like a machine learning problem. We'll see in a minute that it has aspects of it, it has aspects which are not necessarily this. Okay, so the first thing we did, I have a way of doing biology problems which makes my biology collaborators crazy, which is that it's very hard for me to read all the biology papers on a given subject. If you go and say, oh, I'm interested in neoantigens and how they're detected, if you type that into Google, or these days you type that into your favorite AI system, you'll get back many, many hundreds, thousands of papers that you should read, and I guess you can trust some AI to summarize them for you, but that's a dangerous maneuver. So the first thing we typically try to do is try to make for ourselves a very simplified toy model of what's going on. It's not so much that toy model is going to be directly relevant to explain experimental data, but it's going to give us some conceptual framework to think about the problem that then we can go and see if that makes sense, and then we can decide how to generalize from that in order to begin to make more quantitative products. So it's a multi-step problem, and we get referee reports that says, but this model is not motivated by direct biology, and the answer is that's part of the point. The model is motivated by being solvable, because then we can learn the lessons from it, and then we can go on to models that are motivated more directly by actual biology. So that's what our approach. So initially we tried this already some number of years ago. We created what is a familiar idea in the protein interaction, protein folding field. I'm not going to describe it in detail, but it's called random energy models. This was one of the tools that was used for people studying protein folding. It's basically you give up on all the molecular details and assume the interactions are random. Now, when you assume they're random, you get nice Gaussian distributions. You can solve problems exactly. And so we did that. So we created a random energy model, and I'll tell you a little bit more about that in a second. But the random energy model taught us a very, very interesting lesson about how you should think about this negative selection, the training that the system is undergoing. And I like to give an example of this in a sort of joking way. So the joking way I like to give an example of how you can think about this is, let's say you're having a party, you're a graduate student, and you're having a party, and you don't want to invite other physics graduate students to your party, because you think that they're not going to be excited. Or like, other students, other departments, math, chemistry, biology, but you don't want physics graduate students. So you're going to look through the list of students, and you're going to say, how do I know if someone is a physics graduate student? And there are two different strategies you can adopt. You can make a detailed study of the characteristics of physics graduate students. Whatever, the way they dress, the way they talk, how they relate to the world, etc., etc. You can make those would be features of physics graduate students. You can learn all those features, and then you can apply a filter based on those features to any student that you come across. Decide, oh yes, they're a physics graduate student. Oh no, they're not a physics graduate student. This is how machine learning used to work before deep learning sort of took it over. But this was sort of how machine learning used to work. Is you found features that corresponded to the data you were interested in. Features that say this is a cat and this is not a dog. So the problem So that's sort of indicated in the top figure up here, that you have, cases, and you sort of draw these yellow regions which define regions of features that are common that are going to be excluded. Now, if you do that, you make a certain type of error. The error you make is overgeneralization. Those features could apply to students who are not physics graduate students. They might be chemistry students who sort of, have enough of the features of physics students that they look similar, and you would reject them when you should not be rejecting them. That's one type of error you can make. The other choice is to go and sit in the physics department for a week, take pictures of all the physics graduate students, memorize their names, and then delete them. So you just have a memorization process. You don't try to generate features. You try to learn who all the physics students are, and then you just delete them. The problem there is you make the other type of error. The other type of error is where that person didn't happen to show up in the physics department that week. They're on vacation or whatever, so you don't have their picture, you don't have their name, and so now you admit somebody who should have not been admitted. So you make a different type of error depending on what is your way of classifying individuals. So it's the same thing for these peptides. You can say there's some features of the human peptides that somehow allow us to define those things, and that if you're not in that class of features, you reject it. Then it's the opposite. Then you accept it. You don't die. You go out and be released into the blood. Or you can just say that's not possible. You just memorize all the human peptides, and then you go and those are the ones you delete, and everybody else gets accepted. And it turns out that in our model, and also based on our analysis of experiments, very much it works the second way. It works in the anti-machine learning world. It works by memorizing. In the machine learning world, that would be called something like overfitting. So it's the opposite of what most machine learning things would do, but that seems to be the way it works here. There's evidence is from our model, one can see that the advantage of overfitting is that one amino acid change is enough to give you a detectable difference because you're memorizing the sequence. If it's not one on your list, you don't recognize it. One amino acid is enough, so you actually can show in our model that that allows you to do a pretty good recognition of tumor neoantigens. There's also another paper, which is that who tried to apply an information theoretic approach to this problem and tried to say, let me look in detail at all the proteins fragments as part of the human. Let's look in detail at all the protein fragments as part of viruses, malaria, other things. Can I find features? I mean, it only works if there are such features. And in an information theoretic way, they showed that they could not find any such features. This paper is a funny paper because it was originally posted to the bioRxiv about 3 or 4 years ago. And so it's listed here as archive 2025. This paper was actually done about 3 or 4 years ago. It's still under review. The Levine in that paper is not me. It's actually my son. So it's sort of an interesting half my genome. And then the other consequence of this is I told you what type of errors you could make. If there are no features, if you're just memorizing and you only get to memorize 10 to the 4th, 10 to the 5th peptides because that's all you have time to do before you have to leave and make your decision, then you're going to make some mistakes. You're going to release T cells into the blood that actually can detect human systems, and therefore those then you do see those type of errors, and that's part of the reason for these higher levels of testing that keeps going on and on, that it isn't just enough for the molecular recognition, you need cellular recognition, you need things at the tissue level, etc. Okay. So, is machine learning, so now we've sort of defined the problem, and now one can ask the following question. Maybe this whole idea of trying to understand this in detail maybe is not the right way to go. Maybe machine learning is the answer. So, machine learning has an interesting history. This is my version of the history of machine learning. So I was working for a company in this period of machine learning. This period was called expert systems. For those of you old enough to remember that, expert systems was where you were going to analyze some data. You put an expert in a room, and you sort of learned all their secrets, and then you tried to program those secrets into a computer directly by the various computing languages. And, it had a boom. It went up, and then it sort of died because it didn't really work. And there was even an earlier boom. I wanted to point out this very nice quote. If you read the quote, it sounds like something that would be written today, in any type of AI context. And now in the world, machines that think, learn, create, machines will decrease rapidly. The range of problems they can handle will be coextensive with the range of the human model. This was written in 1958, just to give you an idea. So whether this is the time where we really see this coming true or not, it's really hard to know. But anyway, you can certainly try this for any problem you have. And the problem is people have now tried to justify large language models directly to this problem. And my opinion is it doesn't work. So there are many papers. In fact, when we submitted our paper, which you'll see in a moment, is not based directly on a large language model. We sent it in and then got referee reports 3 or 4 months later. And one of the referee reports said, well, you didn't cite the following 3 or 4 papers that have come out on this subject. And actually, those papers had come out between the time we had submitted our paper and the time we got the referee report. This is how easy or how many people out there are just trying to apply large language models to sort of everything. Now, so if you look at what people in the literature are saying, and we agree with these statements, the sequence-based approaches can predict, things that are close to the training data, but they not yet prove the ability to generalize, to be able to do things that you haven't seen. It really doesn't work. The more detailed study of this is the paper published in Cell Systems this past summer, comprehensively mapping all the targets of the T-cell receptor. This is challenging because we just don't have enough data. We just don't really know how to do this. Okay, so we don't do that. What we do is some mixture of molecular biophysics and machine learning, maybe old-fashioned machine learning. I won't go through the details, but essentially what we do, and this is written up in 2 papers published over the last couple of years, what we do is we say, okay, we have let's say, an unknown T-cell sequence, an unknown peptide. We need to create a structural model. We need to know what is the structure, at least approximately. So we have a series of templated structures, cases where people have crystallized this complex between the receptor and the presenting part of the other cell. And we had, initially, we had for our conditions, we had like 70 template structures. We decide which structure looks like it's the best match to the existing unknown structure of the pair being given. And that's done purely on the basis of sequence, either trivially or there are more sophisticated methods for things like TACR distances, things like that. And then once we do that, we can create we have a structure, and then we can evaluate some of the binding energy based on sort of more traditional molecular biophysics techniques. And what we do essentially is train a matrix based on known answers. So we take known crystal structures and create structures. We take known strong binders and create an energy matrix of how strongly amino acids that are really in contact physically based on the structure at the molecular level. We can we can assign energies to those things based on a learning procedure, and we can evaluate what the energy is and make a prediction as to whether this thing looks like a strong binder or not. So it's a mixture. Obviously, it's based on data, it's based on learning information from the data, but it's placed in the context of really trying to evaluate the energy using molecular biophysics. And again, these ideas have come to some extent from the protein folding and the protein-protein interaction field in order to do this. You can do pretty well, so I'm not going to go through the details. I mean, we think we do reasonably well, with some caveats that we'll talk about in a second. So here is an example of one. So if you look at the bottom one, probably is the best one to look at. Here is an example where this is an easier test than just the absolute detection or not. This is where we have a series of possible T cells that bind to something. And we want to say, okay, we have 5 peptides, we have 5 T cell receptors. We know that they bind in pairs because that's what experimentally was determined. And we ask the question of, given the TCR, which one of those peptides is associated with that TCR? So it's sort of a 5-way classification problem rather than just an absolute does it bind or not bind. It's which one of these peptides is the one that binds to this TCR. It's an easier problem to look at, and for those types of problems, we do very well. So the correct answers are the blue ones, the incorrect answers, and if you see what and this is a graph of our energy calculation and binding energy calculation, and you see that the correct answers clearly separate from the incorrect. So we can do this task reasonably well. Here's another task that we can do reasonably well. It turns out that for some particular peptide, 1E6 TCR, peptides which bind this, there was a group that did a full-scale mutagenesis of the peptide. They took the peptide 10-mer sequence and they did a full mutagenesis. They developed tens of a huge number of different sequences. They asked which ones bind, which ones don't bind, looking at exactly how often changing one amino acid in the peptide reduces the binding of something that was a known strong binding. And again, we can make a decent prediction based on that. So things that are reduced tasks that are simpler than the full task, we can do reasonably well on. Now, if you ask, when do we do reasonably well? We've been asked in our model, related models, related to the molecular biophysics motivated models, and ask, when do we do reasonably well? The answer is, we need to have a good structure. So we can do well when we have some reason to believe that our hypothesized structure for this pair is something that's a pretty good guess. We don't know the structure. It's a new pair, never been measured, and maybe it can't be measured, maybe it doesn't bind at all. And so if we can convince ourselves to have a good guess for the structure, then we can actually do a reasonable job of predicting if it's going to bind or not. If we don't have a good guess for the structure, then we just don't do it. We just don't know. We just can't come up with the right answer. So here's a graph sort of showing that statistically, the similarity score is how similar this example is to one that we sort of already know a structure for. So it's sort of a measure of how confident we are in our assigned structure. And this, on the other axis, is what we predict for the binding of a strong binder. And the answer is it very strongly correlates with our ability to make structural predictions. So that, of course, brings us to the question of, well, maybe using large language models is a long machine learning approach. Maybe we just need an approach which can generate new structures. So what approach can generate new structures? Well, the answer, as already mentioned, the Nobel Prize last year in chemistry is AlphaFold. In particular, there's a new version of AlphaFold, it's AlphaFold 3, which actually does a pretty good job of generating structures. And I wanted to mention, I have a sort of interesting historical context for myself, that, in 1988, I was a new faculty member at UC San Diego, and there was a physics talk given by Terry Sadowski. Terry Sadowski, I view as, in some sense, he lost out because he could have been part of that Nobel Prize in physics for developing the machine learning ideas from statistical physics. And so he gave a talk, which was the first talk I ever heard about using neural network models to predict something in biophysics. And he was predicting protein secondary structure. So it was a physics talk, and I came out of that physics talk and saying, this is just crazy. I mean, its results were only slightly better than chance. And he wasn't using any information about molecular biophysics, about physical chemistry. He was just trying to learn from example. And I said, why would you do that? Why would you only learn from example when you have all this knowledge about how amino acids really interact with each other and how helices and data sheets and all these other structures, how they actually form? And so I was very unimpressed. And of course, history has proven him right. Of course, it took years. But nonetheless, I guessed wrong, and of course we know that now you can do that essentially by learning from example. Now, if you ask why that happened, just go back and ask, how did AlphaFold get to where it is now? It really does work at some level compared to 1988, where I was convinced it didn't work. And I check marks of my own bias, but these are the four things I could think of about what's changed since 1988. Some are obvious. So, for example, order of magnitude's increase in computational power. Okay, that helps. Obviously, analyze a lot more data and do much more significant computations. We do understand better the basic physics of proteins and amino acid chemistry and all that. But the real answer here is creation of large databases of actual crystal structures. If you go and look at the database of crystal structures, which is called Protein Data Bank, you will notice that the rate of increase of the number of structures in that system has gone up. I don't forget how many So we decided to try this, and the answer is it does work at some level. So if we say, okay, we have a measure if we believe the structure that we imputed from looking at neighboring things, and we have a measure as to whether we believe it or not, whether that's a good guess or whether it's just randomness. So right now what we're doing is we say if that quest is not good enough, we think that that's not a good approach, we try to generate a new structure using AlphaFold. AlphaFold also gives you information about whether it thinks the structure is good or not. So you can use that as your decision about whether you're going to use your simplified threaded structure from known structures, or whether you're going to take this ab initio computed structure. And I don't want to talk about this in detail, but the paper by my collaborator goes through in detail and shows that you can improve your performance. So, for example, if you look at the red curve, this is measuring, binding some particular test, and you notice that on the right-hand side, where you add these additional structures, your ability goes up in terms of prediction by using computer-generated structure as an adjunct to the known structures in the system. And that's the direction which we're still trying to go in the future. So the summary is, this is, as I tried to explain to you, this is one part of a large problem in understanding how the immune system, which is a very complex system that has to act in an intelligent way, tries to figure out if a cell is worth attacking or not. And that decision can have very severe consequences. If it makes the wrong decision in one direction, you can die of a viral disease, or you can die of cancer. If it makes a decision in the other wrong direction, you can attack one of your own tissues and get diabetes or inflammatory bowel disease, or a whole list of autoimmune diseases that correspond to your system, the immune system attacking. And the decisions continue. That's a sort of molecular decision. Decisions at higher level also have consequences. Many of you might know that in the 1918 flu epidemic, where many, many millions of people died, one of the primary causes of death was not the flu itself, it was an overreaction of the immune system. The immune system went into this overexcited state and never didn't go back to normal when it should have, and the immune system essentially killed the people who would otherwise survive the viral infection. One way you can see that is that unlike most other diseases, viral diseases, in the 1980 flu, the sort of the younger and healthier you were, the more chance you had of dying, which is sort of quite remarkable because it was an overactive immune system that actually killed many, many of those people. So it needs to make these decisions. We've been trying to understand this at various levels. At the level of the molecular decision, the community is making progress. Sequence-based approaches, again, 40 years from now may solve a problem. Maybe 20 years from now, as the speed of progress speeds up, I probably won't be around to see it. But nonetheless, maybe I will. I'll be here at the 28th Granada Seminar. But right now, if you want to make progress on this problem, you can't really use a pure sequence-based approach, but you can use combinations of structural biology, molecular biophysics, and machine learning to still make significant progress in some aspects of the problem. And we're currently continuing to investigate how we can push them out of those interactions. And more generally, in general, I don't understand why you sometimes believe that it's better to not incorporate existing knowledge about biology, machine learning. But I again could be wrong about that. Maybe I get that argument with machine learning. I say, oh, you bias your answer. Yeah, true. It's good to bias your answer. So that's another philosophical discussion. Maybe other people can talk about machine learning with motivation. Okay, so I'm done. These are the people who work on the T-cell that I discussed. Jason George actually is an ex-student of mine who's now a faculty member at Texas A&M Bioengineering, and then Jin Lin did this work postdoc at MIT. Now he has his own faculty job in North Carolina. Wu Wang was my postdoc, who did a lot of the technical work I described to you. And interestingly enough, she went to work for industry of finance, microfinance. That was Eric space centers continuing on with our profession. And thank you to other people collaborating with Michael Markoff as an Indianologist on our team for helping us check out that. Thank you very much.
So I wanted to ask, wondering, in these questions of awe and binding, does ever kinetics come to the picture? For instance, when the property of binding can also be quantified by reaction rate or environment that cells create around them.
I was in the same direction. So I have the feeling that somehow kinetics is important because affinity is easy to measure and on and off rates are hard to measure. But if you compare how machine learning does things, you use huge amounts of data, millions of entries in a data set. Biology with just 10,000 in the thymus is able to do that. So maybe, I don't know, the solution that the evolution has come about is using kinetic information, not just on and off. So I think biology is not binary. I think it's more a continuum.
Okay, so there's one more question.
Just a very future question. How far we are? What do you think about how far we are right now to get an effective cancer vaccine based in this research?
Okay, so okay, so I think we should move on. Let's thank Professor Levine again for a nice talk. So, okay, our next speaker is Professor Belousov, who is going to talk about microcanonical ensemble out of equilibrium.
Yes, it's here. So, can you hear me like that from the microphone, right?
Very well. So thank you, organizers, for giving me this oral presentation. I also, during the presentation, welcome questions right on the presentation, because my goal is to present a theoretical framework which might be useful maybe in your research for some of you. So I would be glad to have some interactions, even if I have to sacrifice some slides for that. What I'm presenting today, in short, is a way to drive microcanonical ensemble out of equilibrium. I believe that there's no need to explain to this audience how important and foundational ensemble theory is for statistical physics. However, I need just to recollect a few developments that give some momentum to my story. I would start with Gibbs. As you well know, it's through works of Gibbs that ensemble theory was formulated as a comprehensive framework of statistical physics. In particular, on the slide I reproduce his formula for the entropy. Here, S sub sigma is the Gibbs entropy. Sigma is a phase space, the space of all possible states that the system is allowed to take. And if you have some probability distribution in this space, you put it in the formula and you get a number. This quantity gives entropy. Then James observed that, in fact, all the ensemble distributions can be obtained by considering Gibbs entropy as a functional. So if we maximize this functional with respect to distribution functions subject to certain constraints imposed through Lagrange multipliers, we recover all distribution functions of the ensemble theory. For example, if we constrain the system to have a certain energy, the Lagrange multiplier turns out to be the inverse temperature, and we recover canonical distribution function. About the same time, James and I should also mention two Soviet scientists, Virukov and Karpov, were contemplating the idea: what if we can define an entropy of a trajectory? By trajectory, I mean a succession of states that the system is going through in the course of its dynamics. Then James coined this term, caliber, which is just a generalization of Gibbs' formula for distributions over trajectories over some interval of time t. However, the question now comes: what should we put as the constraints of the system? In fact, the answer to this question is the same as knowing what are the distributions over the trajectories. Where do we take these constraints? One idea was to impose some phenomenological laws of non-equilibrium thermodynamics, and probably the most appealing was to impose as a constraint average flux through the system. These ideas received sufficient consideration in literature, and at the outset, applying this principle renders some effective models that we can apply to recapitulate some macroscopic observations. However, the research on systems in which we have access and can peek at the realizations, so at microscopic realizations of dynamics, we typically discover that predictions of caliber formalism do not hold. Exceptions can be found in equilibrium systems, but for those we know what are the constraints from the ensemble theory, and maybe systems very, very close to equilibrium, or as an example, fluctuations inside equilibrium states. What I mean, for instance, a Brownian particle that explores medium, and that medium is globally in equilibrium. So to seek further insight about caliber principle, with my co-authors we turned to original developments by Boltzmann, namely microcanonical ensemble, and Gibbs acknowledged that himself that theory of ensembles, at least at the level of microcanonical ensemble, was already known to Boltzmann. Boltzmann himself called it monode. And then we turn to the definition of entropy following Boltzmann. Boltzmann defined this quantity for a macroscopic state and related the entropy to the logarithm of number of realizations of the system state. Then assumption of equal a priori probability of these microscopic realizations connects us directly to the information theoretic framework and the framework of Gibbs entropy. One of the goals for Boltzmann was at his time to connect fundamental laws of physics. At his time, it was still a hypothesis that matter at microscopic level has a structure, molecular structure, and therefore he chose a particular set of phenomenological thermodynamic constraints that had also been understood through classical mechanics for particles following Newtonian motions, Newtonian equations of motions. We know that energy is conserved, mass is conserved, linear or angular momenta are conserved. That cannot be said, for instance, for the average flux. Nonetheless, through Boltzmann was successful in establishing equilibrium statistical physics, so maximizing Boltzmann energy, so the state of the maximum Boltzmann entropy corresponds to equilibrium, as you well know, and in particular, byproduct of it, that we have also established canonical distribution, the Boltzmann, Maxwell-Boltzmann law. But now I'm asking, can we actually use similar approach to count number of realizations of trajectories? Okay, for instance, if we take very, very tiny interval of time, can we count realizations of a trajectory? Then we go a bit deeper to developments of Boltzmann. In particular, he himself used in his papers this model of ideal gas, which we can call Boltzmann gas. We consider a box filled with particles. Originally, Boltzmann considered them to be distinguishable, but allow me to skip the developments of the later years. Today, we consider usually indistinguishable particles, and particles must be distributed over m energy levels, possibly degenerate with some multiplicities. And then we want to count how many ways we can distribute particles over these energy levels. This combinatorial problem is known, and we know how to solve it. In particular, I'm reproducing here the formula for the logarithm of number of realization after application of Stirling approximation. Now I'm going to ask what can happen in the system if we allow it to evolve in time. Okay, one simple idea that this is perhaps the simplest thing, we consider a discrete evolution of the system, and we just allow particles to redistribute themselves. And we allow absolutely any kind of transition. So particle is free to start from a certain level and go to any other level in the system, or if it wants to remain in the same energy level where it starts from. So can we count this number of realization? In this case, to specify a trajectory, that means just to tell how many particles we move from one energy level to the other. And what I suggest to you, just to focus on one energy level. For instance, here in my slide, I'm
focusing on particles from the source energy level epsilon 2. So what we do, we take these n particles and put them wherever we want. That's exactly the same problem that Boltzmann already solved. We just need to apply it to all energy level and sum up. So the formula turns out exactly, almost exactly reproducing the steps of Boltzmann development. Here, n sub ij, that's the number of particles we moved from energy level j to energy level I. But to set up the stage for non-equilibrium effects in the system, we need to get some more freedom in design. And one aspect I want to bring up is that typically when we deal with ensemble theory, we consider a system as a box. And we quantify thermodynamic state by temperature energy for the whole system. However, as soon as we want to go to non-equilibrium theories, we have to allow these quantities to vary in space. So we may have different energy, different number of particles in different parts of the system, so that gradients of temperature, gradients of energy can exist. This situation can be easily mapped to the idea of lattice gas. So allow me also to apply here a periodic boundary condition. So we think of our system as consisting of several sites, or similar to histogram bins. Each site has its own energy levels. And periodic boundary conditions closes the system so as to mimic infinite extensions in space. In general, of course, we could allow these structures of energy levels in all bins be individual, but for Boltzmann gas, perhaps you would agree, the simplest thing is to assume that energy levels are the same across all bins or sites. Next idea is that we can actually design our dynamics with more constraints. For instance, a reasonable would be to expect that particles are allowed to be redistributed within the same bin, or to go in some neighborhood, for instance, nearest neighborhood, or nearest neighbors, or second nearest neighbors. In my talk, I will reproduce some results, always assuming only nearest neighbor moves. Although energy levels now should be labeled by two indices, it is just bookkeeping. In fact, if you have finite number of energy levels, you can easily flatten this index into one. Very little is changed for the formalism. And here's the most general expression I'm going to show you today of the microcanonical caliber. So we apply summation over possible transitions from levels L sub I, source energy levels, to energy levels Mj in the M bin at the level J. And to specify uniquely a macroscopic trajectory of the system, we just need to specify numbers of particles moved between these energy levels. And now we can think how to maximize this object. So we are going to maximize the objective function, which is composed by microcanonical caliber, log n omega, and Lagrange multipliers coupling the constants. What constants could we apply? So here again, the Boltzmann developments, they allow us to relate these models to fundamentals of physics, and you would probably agree that one next to their energy mass conservation, what we can more fundamentally understood constraints are continuity equations. That's, for instance, as opposed to average fluxes. So we know that if particles are not allowed to be created or annihilated, then we cannot move more particles from energy level ij to any other energy levels in total. So if we sum over all target energy levels, we should obtain the number of particles we had originally in our energy level. That is discrete analog of continuity equations. We could also impose energy constraint, that is the same energy constraint that Boltzmann used. Again, it is well grounded on the classical mechanics. Or if you are interested in energy transport, we could even cast energy constraint as another continuity equation, and then we could study the transport of energy, for instance, heat flows. And in principle, this family of models that Boltzmann started is very flexible and versatile. In fact, today there is a large range of models called Boltzmann lattice models that used even to address hydrodynamic problems. So for equilibrium dynamics, I just briefly mentioned, typically we obtain the dynamics in the form of Markov chain. That is also true for out of equilibrium generalizations that I'm going to consider. This in Boltzmann gas Markov chain, this Markov chain obeys detailed balance condition, and it actually directly leads to Boltzmann distribution of equilibrium in the steady state. However, through this approach, we can obtain even more general expressions of detailed balance, for instance, considering interactions, non-trivial interactions, for instance, Fermi-Dirac statistics and exclusion principle. And while I'm working in biological institute, I'm not that much referring to these exclusion principles in the quantum sense. Often we use such approach to model steric interactions. However, down in the slide, I'm reproducing steady state detailed balance for Fermi-Dirac. For transient states, we would obtain even more general solution. To best of my knowledge, it did not happen to appear in literature so far. Now, to the main dish of my talk is how to drive systems out of equilibrium. There are several constraints that we could motivate. For instance, more traditional systems studied in non-equilibrium thermodynamics, covered in classical books, that could be cast through constraints known as Norton ensembles in the literature. What we do, we close the circuit. So here I depict a schematic of a simple system with 3 sides, and just the terminal sides, first and the last, I constrain so that there is in each step a net current of particles between them. That could be implemented, that could mimic a working battery, or if you like, a Maxwell demon that does it for us. However, inside, in the central bin, we would observe no direct consequence of, we would not observe direct constraints, and dynamics in those bins would reproduce something more natural to the system. While we implement this constraint in dynamics using Lagrange multiplier we call current affinity, we could also consider what is called equivalent ensemble. Equivalent to the Norton ensemble would be Thevenin ensemble, in which instead of fixing the flux, we first derive the equations, and these equations would contain current affinity, and then we actually fix the current affinity. So the difference is that the Norton ensemble current affinity must be a fluctuating quantity so as to impose the constraint. In Thevenin ensemble, it is just fixed. Other maybe more interesting to the area of my research, types of ensembles, active ensembles. So we often observe that in, let's say, bacteria or some other active matter, even synthetic active matter like Janus particles, they have ability to usually consume some fuel and to propel themselves. One model that we can motivate with this from these systems is a directed motion. So we can constrain particles, always a certain number of particles in the whole system, always to skip a bin. And because this is a global constraint, this is also parallel to what is used in traditional ensembles, because if we divide this number by the length of the system, that roughly corresponds to the average flux. Another interesting example is active diffusion. So we can actually force particles to move, but we don't care about what direction. So the total number of particles must be displaced in one dynamical step, but they could be displaced left to right, and we do not care about their direction. Let's see some examples, because one apparent feature of this theory is that we can simulate these systems. So I literally simulate here a three-side lattice. And I'm going to ignore the energy structure, actually, just to survey some statistical features which are interested to matter flow. In equilibrium, of course, as you would expect, we would obtain a flat distribution, nothing new there. But if we consider Norton ensemble and impose a flux of 100 particles in each step between bin 3 and 1, we can measure current that of particles passing through the middle bin. In this system, we actually find out that this external constant generates a gradient. That's precisely what is discussed in a classical non-equilibrium thermodynamics textbooks, for instance, wrote in Mazur. Then, in this system, when we simulate, we can also measure how the current affinity fluctuates and measure its steady state value. What we do then, we take this value and put it in Thevenin ensemble. What we discover is that Thevenin ensemble generates precisely the same statistics of particle distributions and currents. In the directed motion, we absorb current, but as you can see, we don't build up a gradient. That's because the system is symmetric, and we all observe that particles are just, there is no difference between bins, and therefore there would be no macroscopic gradient. In active diffusion, we observe flat distribution of particles, no current, but we know that this is non-equilibrium system. Because, for instance, we could now talk about the statistics of the current in the middle bin. Of course, in equilibrium, it is zero with some fluctuations. In Norton and Thevenin ensemble, the statistics of the current is the same. Interestingly, the amplitude of fluctuations of the current are the same as in equilibrium. These are the signatures of the local equilibrium. So in these ensembles, we also establish existence of local equilibrium condition. Interestingly, in directed motion, we can tune the parameters of simulation so as to have the same average current, but we also observe suppressed fluctuations. These are consequences of long-range correlations in the system. So if we motivate this system by the idea that particles need to consume fuel, which is concentrated within the system, so if we know that a certain number of particles have moved from a given bin, we know that other particles would not find enough fuel to power their motion. That gives us extra knowledge about total current in the system, even if we observe a single system, single site. These correlations, they actually work out so as to suppress the amplitude of fluctuations. Finally, in active diffusion, with enhanced diffusion, we uncover that although the flux is on average zero, its fluctuations are increased. That these are the phenomena which typically modeled by effective temperature and correspond to increased frenetic activity. And even more. So there are more things that we can do with this framework. So just some examples we already have where we found useful. This framework presents some studies in which we were interested in dynamics of transients with my co-author Jenna Elliott. We could generalize some frameworks which typically rely on assumptions of local equilibrium, but we can actually remove necessity to invoke equilibria assumptions by going to the caliber principle and making the dynamics fully non-equilibrium. We can now treat actual emergence of inhomogeneous transport. So from these models, we can reconstruct Swolkovsky diffusion equation, and in this diffusion equation, transport coefficients can be related to the connectivity of the sites and energy structure of the levels. Finally, while I mentioned only generalized detail balance condition, this framework by straightforward calculation gives you generalized Markov chains with a prescribed broken detail balance that is a consequence of the imposed non-equilibrium constraints. We are currently working also on applying these models to heat flow, because now we can treat in a principled manner heat flow problems using random walks, and as far as I know, this is quite novel development. I'm summarizing here the main points about this approach. First of all, in difference from the traditional caliber principle, which you can find in literature, we are considering here local constraints in time. This yields physically plausible dynamics, but yields somewhat more complicated distributions of trajectories, which you come up with some equations, and if you solve them, you can find the distribution of a trajectory over a longer time interval. Connection to continuum models is also quite straightforward. For those of you who are familiar with random walks, would immediately recognize that taking continuum limit, we can go to Poissonian process from Markov chain and to field theories through by taking continuum limit of the lattice. Finally, I want to mention and emphasize further that dynamical ensembles with microscopic constraints can be tailored to physical properties of interest, although Boltzmann developments may look very toyish in principle. These models have a lot of freedom, and I believe that the previous speaker was also would confirm that one can come up with quite realistic concepts with these approaches. I thank you for your attention.
So how much testing has there been done just at the level of looking at probabilities of trajectories, which was the original thing that Max McCollum was supposed to, or the generalization with different constraints? I mean, is there some studies that basically says you create some microscopic model, run many simulations, discover the statistics of the trajectories, let's say between in multi-stable systems, whatever it is, and see to what extent it fits the, whichever version of Max-Cal or constraint. So so maybe just give me, what is the status of that?
Yes, so this we have a preprint published already about this story. So far, best to my knowledge at least, there are some similar developments for diffusion problems, and as far as I can see, through our work, we can recapitulate so many features which happen in real physical systems. So here the question is more like, if you have some physical insight about the system, you always can find well-motivated fundamental constraints translated in the language of random walks to recapitulate them. So, so far, even, well, now it's in progress, the heat flow problems, we were always able to find steps that produce statistics comparable to real systems.
So in the biological world, there are many models of, I would say, chemical dynamics, more than diffusion, whatever, just interactions of networks of molecules that then have different states, and one is interested in trajectories that go between different states, or what are the probabilities of those trajectories? Are there multiple trajectories, different probabilities? So this could be applied. So the answer you gave is this could be applied to those sort of directly.
Yes, indeed. That's exactly why I want to share these ideas, because indeed, just the systems which you were describing in your talk, I think could be examined through the lenses of microeconomical caliber, and maybe come up with some kinetic aspects to the things that you model in your papers. Yes.
Okay, so if there are no more questions, let's thank the speaker again. So we continue now our session with Professor Ana Paula Millán from the University of Granada, who's going to talk about how topology shapes dynamics of higher order networks.
¿Me lo mandaste este correo? Esperando que...
Abre el correo.
No, pero tienes conexión para conectar. ¿Con qué? ¿De USB-T? ¿USB-T? ¿Puedes preguntar la nombre aquí? ¿Qué es un PowerPoint?
Mándalo al correo.
Lo que tú quieras. Mándamelo por correo. Sí, se van.
Okay, so now yes. Topology shapes dynamics of higher order networks. Thank you.
Okay. I think this works. Yeah. Okay, good enough. Thank you for the patience. Yeah, so I want to present. Please see, I think this is quite taller than me. Some recent work that we've been doing on the general framework of dynamics on higher order networks, and mostly talk about two recent works, which are synchronization of higher order remote models and triadic interactions, which both give us ways to allow for the dynamics of the links, or let's say the connectivity of a network. Starting with synchronization, here I don't need to explain what that is, but instead of focusing or talking about the traditional synchronization of signals associated with the nodes of a network, we can also think about the dynamics of the links. This can correspond, for instance, to fluxes in transport networks, like the one shown here for fluxes on a leaf, but also any kind of transport network, and of course in dynamical networks like dynamic connectivity in the brain. And these signals can interact with each other, and they can, in general, synchronize. To describe mathematically this type of dynamics, I'm going to do a very brief mathematical introduction of simplicial complexes that allows me to define the Hodge Laplacian, which is going to be the basis of the model. So these simplicial complexes are generalized network structures where we can encode not only interactions between pairs of nodes, but more generally between any number of nodes. And actually, we not only allow the nodes to interact, but also the links can interact among themselves through the triangles that they form. And the triangles can also interact, forming tetrahedra, and so on. We we can keep increasing the dimension. Why simplicial complexes are a particular type of hypergraph, and that has a few extra conditions. One of them is that if we have an n-dimensional interaction, so if we have a tetrahedron of the dimension 3, all the lower dimensional simplices or interactions are also included. So if we have this tetrahedron, all the triangles are also part of it, all the edges are part of it, and also all the nodes. So we cannot have, for instance, the situation where 3 nodes interact, but we don't have the pairwise interaction. This is a restriction that gives a lot of topological properties to the systems that we can then exploit. One of them is that we can define the boundary of a simplicial complex, and this boundary also belongs to the simplicial complex, and we can derive it through these boundary operators, which basically we can define them on n dimension, and it will take us from dimension n to its n-1 dimensional boundary. Here is a simple example. This is a simplicial complex made by a triangle, these 4 links, and these 4 nodes. And we can define 2 boundary operators. The 2-dimensional boundary, which gives us the boundary of the only triangle. This is B2. And the boundary is written in terms of the 3 links that make the triangle. And we can also define the 1-dimensional boundary operator, which gives us the boundary of the links. So it takes us from the 1D space to the 0-dimensional space of the nodes. And of course, with the co-boundary operator, so the transpose of these ones, we can move upwards. And we have the nice property that the boundary of the boundary is always null, either if we go upwards or downwards. And this means actually that with simplices or simplicial complexes, you don't really encode higher dimensional interactions between nodes. So if you try to see what is the 2-dimensional or the 0-dimensional boundary of this triangle, it's always going to be 0. As we will see later with the when we define this Kuramoto, this higher order version of the Kuramoto model. So another property of these incidence matrices or boundary operators is that we can use them to write the network Laplacian. So just by combining them. So here's again a simple example of a simplicial complex made of these 3 triangles, also these links and these nodes. So with the Laplacian, we can codify how the nodes interact among themselves, and they do so between the links of the network, right? So we recover this interaction by coupling the boundary operator. So we first go up and see which nodes interact through which edges, and then go down to project back on the node dynamics. This can be generalized to any dimension, and we arrive at what's known as the Hodge Laplacian, which is going to tell us how signals that live in dimension n, so let's say on the links, for instance, interact. And now we have two terms, bit of a spoiler. So we first have the same term that we have for the nodes. In the case of triangles, this form, in the case of links, this form triangles. And their interaction is described through the two-dimensional boundary operator. So this is the direct extension of this term here. It's a very similar type of interaction, but now instead of pairwise, because a link always connects two nodes, we have a three-point interaction, because a triangle always is going to connect three links. But of course, we have another option now, which is that links can also interact through nodes, because they can connect, so several links can join at the same node. And this is recovered through what's called the downward Laplacian, and now uses the the n-dimensional boundary operator. So we project the links to the nodes and then go back up to the links. And this interaction now is different, because a node can connect any number of links. So here we have an interaction, a pairwise interaction between these two links, but here is 4 points, and I don't have an example here, but here we would have a single link interacting with itself. So this term, this downward Laplacian, is going to add really a new different type of interaction into the equations. We have some properties that I won't really discuss in much detail, but the most important for us is that the dimension of the kernel of the Laplacian is the corresponding Betti number in that dimension, and that the Laplacian is orthogonal with L down and L up, commute and can be diagonalized together, and thanks to that we get the Hodge decomposition, so we can decompose any n-dimensional signal into its harmonic, rotational, and solenoidal components, where the harmonic component lives in the kernel of the n-dimensional Laplacian. And the rotational solenoidal are given by the boundary and co-boundary operators. Now, going back to synchronization, which was the main point of our study, we can, if we start from the pairwise Kuramoto equation, where here we have a system of n oscillators, each given by their phase. Each oscillator has an internal frequency. They are coupled through a network A, and we use a simple sinusoidal coupling function. This equation can be written directly in terms of the boundary operator. That again is telling us how the nodes are coupled through the links. And of course, if we linearize the system, we arrive at the graph Laplacian. In the higher order description, we can directly extrapolate, or we propose this definition of a higher order Kuramoto dynamics for the n-dimensional signals by extrapolating this equation, where now we have an extra term. So we have that the dynamics of the signals are driven by their internal frequencies, of course, by the interaction through the upper dimensional boundary. So this would be nodes interacting through links, links interacting through triangles, and so on. And also by the lower dimensional boundary, which is the term that we don't have here. Again, if we linearize, now we arrive at the Hodge Laplacian that is controlling the linear dynamics. We can then study the dynamics of the system, which we did numerically initially, and do your standard analysis of, okay, let's see how, let's first pick a dimension, and in this case we are going to do the simplest thing after the nodes, is the links. We connect them, or we let them evolve according to this equation, and we have the coupling strength as the control parameter of the system. We measure the synchronization of these higher order phases, and we expect that they will synchronize eventually, but as you can see here, they don't, ever. It doesn't really matter how strong the coupling gets. We could not find a synchronized state just with this parameter or with this description. And this is independent, or for the most part, most simplicial complex for more simplified complexes will give this solution. So what's happening here? Where I mentioned before the Hodge decomposition, and if we work this a little bit into the equations, we find that because the harmonic components are in the kernel of the Laplacian, they will evolve independently, each of them according to the proper frequencies that we have given to the system. So basically, the harmonic component of the frequencies, unless there is an eigenstate that is synchronized, they will not synchronize. And this is not general. This is the main difference with the case of the zero-dimensional Laplacian or the graph Laplacian. In general, we cannot assure that a simplicial complex will have a constant eigenstate. Instead, this is only the case when we have so when the whole simplicial complex has a whole, going back to these Betti numbers that are define the kernel of the Laplacian. So in general, we will not see the synchronization of the n-dimensional signals, but we can still observe the dynamics in a bit of a better way by studying what's happening in the boundaries. So we can see what the triangles and the nodes are doing by projecting with the boundary operators. So we define these projected signals, which are basically the discrete call and the discrete divergence of the original ones. We see that the dynamics for these two decouple. So the dynamics projected on the nodes are independent to those projected on the triangles. They are basically given by a standard Klamoto equation, just with a coupling given by the upwards and downward Laplacians. And if we look at the synchronization of these signals, this will synchronize. And actually, by propagating this idea, we can see that the dynamics of the links or original signals also synchronize, but not if we look at the full signal, but if we look only at the solenoidal or the rotational component, which we can do by applying the Laplacians, which apply this sort of topological filter. So basically, in this model, we do have a synchronization transition, but it's not apparent in the full signals, only when we apply these topological filters that keep only one component. This result is, this result is quite general. Here is for a configuration model, but you can replicate it in simplicial complexes that are scale-free or that are based on experimental networks, which I will show in a minute. In the previous, so we can play a bit more with the equation, right? So in the previous version, the coupling was the same in the triangles and in the nodes. So we just had sigma here and here, but we can in general try other types of coupling. They don't have to be the same. And in particular, we can, in a way, mix what's happening above and under. So the triangles with the nodes. So we tested, inspired by models in multiplex networks. We will also have these different layers. We tested this coupling where the interaction through the triangles is modulated by the synchronization of the nodes, and vice versa. The synchronization of, the coupling through the nodes is modulated through the synchronization of the triangles. By doing this, the dynamics no longer decouple, because we have this extra term here, and we go from this continuous transition to an explosive for discontinuous one. That again is only observed on the projected dynamic or on the filtered ones, but not if we look at the full signals. As I was mentioning earlier, this result is very general, and it holds also for different types of simplicial complexes. You can see more details in the paper. So in summary, we have defined this, or you can define this interaction between higher-order topological signals that are not assigned only to the nodes of a system, but more generally to any n-dimensional signal. And in that case, depending on the topology, you might observe that the full signals never synchronize, but their filter versions do. And in these two papers, you can see more details of when we can observe synchronization of the full signals, which is in very particular scenarios only. And now to move to the second part of the talk. So here in the previous model, we allowed the links to interact through nodes, but only the nodes that they shared. Now we wanted to study a bit of a more different case in which the state of a link can change, not depending on the node that it's connected to, but any other node in the system. And these are called triadic interactions. Triadic interactions define this scenario in particular when we have a node that projects directly to a link, not to another node, that projects a connection. We observe this in social networks predominantly, so we know that the interaction between two people is going to be different if a third person is present. We also observe them in ecological networks, where the presence of a third species can enable or disrupt the interaction between another two. And interestingly for us, also in neuronal networks, we can find this type of interaction in different ways. So one of them are axoaxonic interactions, where a neuron projects a synapse directly to the axon of another neuron. So it's controlling the synapses of that neuron directly without affecting the somas of the node. And these are thought to be important for learning in some areas of the brain. And we have another famous example with our tripartite synapses, in which we have the synapse between two neurons and a third cell, which is a glia cell, an astrocyte, that can modulate it. So glia cell or astrocyte can take on many different roles, but in some areas of the brain, they take on this modulating effect directly on synapses of the neurons, so they don't affect the node, they only affect the link. I will not get into detail of this particular case. I will focus more on this system where we only have one type of node. But we recently submitted a study with Joaquin and Gustavo, who are also here, proposing a model for this type of interaction where we can look into what is, in the higher order sense, what is the role of having this interaction between synapses directly, and what type of effect it can have on the system. And we found in this very simple case with only 3 nodes, 3 synapses, and an astrocyte or a higher order interaction between them, that having this interaction extends the dynamical range of the system. So it makes it more responsive to external stimuli that the system without this type of interaction. Now, going back to the other type of triadic interaction, so this type where a node directly regulates the link between 2 other nodes, or perhaps one of the first questions that we can ask is how is this going to affect the connectivity of a network? Because then depending on the connectivity, whatever is happening on top of that is going to also be affected. And so for that, we look at the framework of percolation, which again, I don't have happily to explain here, but just for reference, this is your standard, your phase diagram of percolation on a fixed network. Right? And as we increase the fraction of links on the network, there is the emergence of a giant connected component of increasing size. And in general, we have this random damage probability that acts as a noise in the system that will destroy the links. When we add triadic interactions, we will see that we lose this phase diagram because the system is no longer stationary, because the triadic interactions can enable or activate and inactivate links, and thus change the giant component. I'm going to very briefly present these results of this work by Han-Lin Sanz, who will also be at the conference, and then move on to a particular case of a bit more realistic, where we will have spatial networks. So first, in general, we consider a simple system where there is a structural network. Those are the nodes in blue and the links in black. And we allow for this triadic regulation. So the nodes can either activate links or deactivate them. And apart from that, we have the random damage. The dynamical rule is very simple. We say that nodes in the giant component are active, and they will activate the regulation. And then the links that have only positive regulation will be active, anything else will be inactive. So this node that has both positive and negative, this link that has both positive and negative will be inactive. So will this one and this one with only negative. So in this example, only this link can be active in the next step. Changing the links changes the giant component, which changes the nodes are active, and so that's how we get our dynamics. So what happens here? I had a GIF, but it will not play. So you will have to believe me when I say that if we start from, for instance, this network where initially only a very handful of links are active, sorry, which are the ones in yellow. So the giant component is very small. In the next step, we are going to get to a giant component that spans almost the whole system. So almost all the nodes are going to be yellow, and in the next step, we go back to this configuration. So we are going to have a blinking where the size of the giant component goes from almost 0 to almost 1. That's even in the absence of random damage. Corresponds in this orbit diagram to the case of p equal 1, so probability of random damage equal to 0. And we will have this exact blinking between an almost fully disconnected network to a network where almost all nodes are in the giant component. Which nodes are in the giant component will depend on the coincidence, on the specific realization of the network and on the initial conditions, but the macroscopic dynamics are always the same. As we add random damage in the system, as we add damage in the system, the situation there gets more complicated, and we get this many different states that the giant component or the size of the giant component can take. And only after having a lot of damage, we get to the system that is disconnected. So the size of the giant component is negligible. These results hold for any network that is a small wall. So for random networks or for any type of random network where the nodes are close to each other. Now, this is not the case in many systems where instead we have this spatial network. So the probability of being connected depends on your distance. So only nodes that are close together will have a connection, which can apply both to the structural network and the regulatory networks. In this case, we found that where we can now project the network on the space, and we found that different types of spatiotemporal patterns of the giant component emerge. Depending on the random damage. So we have, if there's a lot of damage, we have these small clusters that will move on the network. If there is no damage whatsoever, instead of having in standard percolation a large giant component that spans the whole network, or in the random network case, this blinking that we had, now we have this funny state, I think, of where stripes emerge. So we have that all the nodes that are active form this one-dimensional stripe, and they will move right and left as time evolves. So here you have a bit of a snapshot of the dynamics. And if there is intermediate and random damage, then we have these weird patterns that more or less take up half the network and go from a pattern and anti-pattern. So the next step, the opposite nodes will be active over time. So we now have a situation where the largest giant component does not appear when there is no damage, but when there is intermediate damage. And actually, when there is no damage, we have giant components or patterns that are relatively small. So this is quite different to the situation that we had either for the static percolation or for the random network case. You can do, we were not able to prove this analytically, but we gave, you can give some arguments for it. And these patterns emerge because we have this competition between short-range inhibition and excitation, which means that you can only have activity next to where you had activity before. Not in the same place, because there is too much inhibition, and functional inhibition wins in this system. There is a functional imbalance. And not too far, because the connections are only short range. So this leads you to only having activity or connectivity at the boundary of the previous step. And we find that these stripes emerge through some sort of surface minimization effect because of this, in the case where there is no damage. This previous study was based only on connectivity. We can put dynamics on top of this to go to something closer to what's happening in these axosonic interactions, which is also a way to study what happens when instead of a global rule, which is looking for the giant component, we go to a local rule, which could be looking for how connected a node is. And by doing this, again, you can find more details in the paper, but by varying how strongly the dynamics follows the structure, so loosely or directly, basically, we can find different patterns of activity. These are the gray maps here. So we can go from some patterns that look similar to those before, a bit more diluted, to, so the active nodes here are in black, sorry, to these very pathological or blinking type patterns, where we go from a fully or almost fully active state with some holes to a state where only the holes are active. And so to sum up our findings, triadic interactions allow percolation to become this dynamic phenomenon where the size of the giant component changes in time. And if this takes place on spatial networks, we can have the emergence of spatio-temporal patterns with complicated dynamics. And there are some conditions for this to occur, of which the main ones are this structural balance between inhibition and excitation and the local connectivity, local or short-range connectivity. I want to thank Joaquin and Gustavo, who are here, and were the main collaborators in these studies, and also Han Lin and Ginés Travianconi, who also participated in most of them. Here are the main references for what I presented today. This is a recent review that we just published that goes over these two areas in detail and also presents a few other recent results on the dynamics of higher order topological signals. And thank you.
Hello, really nice talk. Thank you so much. So towards the end of the first half of the talk, you very, very quickly breezed through some results on C. elegans and humans, if I remember correctly. Could you elaborate a bit more on those?
Yes. So these were simplicial complexes built from connectomes, so from the pairwise networks.
Structural.
Structural, yes. So it's I think the Homo sapiens one was the one by spawns, and the C. elegans is the famous, bistructural in both cases.
Okay, so you grab a structural connectome from those, and then you put these high order Kuramoto.
Yeah. So we found basically anytime there were three nodes that were connected, we put a triangle, and then run the same model on them.
Yes. Thank you.
Thank you. Nice talk. I have two questions. First, what is the Laplacian operator for the triadics dynamics? It is the same as the Lodge Laplacian of the.
Sorry, what's the question for what?
The Laplacian regarding the dynamics in the triadic structure.
Oh, in the triadic case, we don't have a Laplacian, actually.
Okay.
There is no dynamical, well, you can derive an equation for R, but in the microscopic level, there is no equation. You just find the giant component.
Okay.
Yeah.
So it is not necessary to do a linearization process to obtain that phase transition, to obtain that
You mean
that cluster?
here?
Yeah.
Yes, so that's in this paper. Actually, this orbit diagram is from the mean field description. It's not simulations.
Okay.
The one for simulations looks similar. So you get to some mean field equations for R and the fraction of links, and then you can derive the diagram. But Han Lin can give you more details because he did all the.
Yeah, thank you.
Okay, so if there are no more questions, let's thank Anna and all the speakers in this first session. So now we have time for coffee, but before leaving, just one quick message for speakers. So if you are talking in the next session, please, and you don't have sent your PDF, please drop by before going for the coffee. So now we have this coffee break. It's the downstairs in the -3 floor. I think you can take the lift or the stairs, and we will be back at 11:20, so just 20 minutes for coffee. Okay. Okay, it's in the same floor. Sorry. So see you now. Thank you.
Downstairs, down, -3. Okay, you will smell the coffee.
Adiós de Pablo.
Thank you. Very happy to be here. Maybe I would like to check whether I can just plug this
So
HDMI.
what type? You have yeah, with HDMI. Yeah? Okay. The problem before was that we have USB-C and then USB connection. So maybe USB. Sí, viene de mediodía.
Y tengo media orilla. Que llevo viajando todo el mes y no me enteré y pensé que era 15 minutos de la charla.
No, vale.
Ya lo había dado antes, así que estoy viendo. Y si no, hay la pregunta está
También bueno. You wanna see watch Interstellar? This one? So maybe I can copy, you know? Yeah. No, ese es. Sí, a ver, mejor.
Pues lo más que le pasó una vez que ya me da pánico.
And now, para pantalla completa,
Un poquito así.
vale,
Para asegurarme de ver la boca. Yes.
maybe leave it open, no? And now, it should be this. Sí, siempre sí. Just, do you wanna use the pointer? Okay, so let's see.
Let's try.
Yeah, but just to check whether just switching off and on. Never works. Always.
To see you.
Do you have all presentations here?
Yeah, I use okay, so you know where the presentations are? Ah, Enrique, perdona, que te estaba hablando de inglés. Encantado, yo soy Pablo. Encantado. La tiene Joaquín, ¿no? Pues se lo voy. ¿La tienes tú ahí? Pues mira, más fácil. Sí, sí, sí. Sí, sí, vamos a descargarla. Ponlo ahí. Sí, yo creo que sí. Ok.
Solo la interacción.
Vamos a ver. Yo no,
Igual que yo llevo 30 años, más de 30 años con Linux.
sí.
Esto lo dejé.
Joder, ya no sé qué. ¿Dónde está?
Pues ahí lo tienes.
¿Esto?
Abajo, eso es el disco que está abajo.
Ah, vale.
No, para abajo. No,
A ver.
no está, no
En
veo.
este
No veo
equipo, espérate, pero está hecho directo.
estaba ahí, ahí tiene que estar la charla. Tienes que, bueno, tiene que estar aquí, pero abajo, me imagino que está ahí la siguiente. China, espera un momento, que me vale, Granada Seminar, Granada Seminar, el PDF.
Este de aquí. Vamos a hacer copy paste, efectivamente, equipamiento ya, copiar. Bien, y ahora aquí, sí, vale. Esta
Vale, simple.
sí, sí, vamos a verla.
Esto va a ser
A ver, la pantalla completa, yo todavía no la controlo.
y para hacer la enganche a control L, una cosa de esa.
Control,
Que no utilizo un data, y la prueba con control L. ¿Esto? Ah, genial. Vale, y luego encuentra, o sea, control L le tira, es que ya no sé si, mira, vale.
Y a ver la otra, a ver si está aquí abierta. Vale, pues ya está. Vamos por
Así,
sí. Pero vamos a ver dónde está, como este de aquí.
Como no estamos escribiendo,
Se puede tirar, sí,
disco,
se
lo
puede tirar. Pues, exacto. Sí, efectivamente.
Escúchame un vistazo, a ver si
Sí.
ahora que me lo dice, vale. Ya veo. En la primera. Vale. Y ahí apago, ¿no? Cierras todo aquí. Sí, por quitar la transparencia.
Puede ser.
Yo por la tarde, o sea, no hay tiempo.
Yo creo que no ahora miro, no sé qué era el primero. Voy a mirarlo.
Vale, bueno, ya veo.
Okay, welcome back, everyone. So we are going to start the second part of this session, morning session. So our first speaker is Professor Luis Seoane, who's going to talk about topological communities in complex networks. So please, thank you.
Hello, can you hear me? Yes. I usually move a lot, so I will probably get hold of the microphone. Let me know if I move too far away. So thank you for being here. I'm super happy. The Granada Seminars, I hold them very dearly. So it's always a pleasure to come to the city and to have something to tell you. I'm going to tell you something that I think is terribly simple. I've looked around in the literature and I haven't found other people using similar approaches. I'm pretty sure that it exists because everything has already been invented. But at the same time, I think that what I'm going to present is very informative about networks in general. So much so that I'm often thinking, well, I think this is the kind of analysis that you should apply to a network when you're given a new network and you don't know what to do with it. The whole story starts around 4 years ago with Iker Atienza, who was then a really brilliant PhD student. Now he's a brilliant postdoc. And Susana Manrubia, who sent her regards. And we were in Madrid at the National Center for Biotechnology. And they were doing some reports about how well the center was functioning. And we were invited to do something with networks, whatever that is. So we decided to build the collaboration network of the center, which isn't anything new. We have seen a lot of them. This is a summary of all the papers that were published in a 5-year period. This was a special 5-year period because the center had a special distinction from the Spanish government. They gave them some money. They made these many papers. We have these many departments. And we basically built a network. Here it's separated by departments, in which every node is a person and 2 people are connected if within this 5-year periods they co-author a paper together. Right? So it's the simplest thing that you can imagine. We come here and we say, okay, what do? We measure some stuff that tells us how often 2 different departments interact. This is the same network but collapsed by departments. This is the same network but only with the PIs. In the big one you have all the people. In the small one you only have the PIs. Node size is proportional to the number of papers. We measure things like centrality of different nodes to see what department was more or less important. We did some clustering and we found that there are some departments that span only one cluster more or less, some exceptions here. This is infrastructure. So these are people that provide like proteomic services. So they collaborate with everybody somehow. So it was a little bit like, this is the network. What can you extract from that? And I was feeling unsatisfied. I was like, okay, is there a way that the network is going to bring you to what is important to understand the network? Like the network might automatically guide your attention to the relevant features, because what we were doing is we're just looking at the structure that we know, which is departments, and we're just measuring a couple random things that we think might be important. But what we were hoping is to get something like, I use this Pollock. There is this very beautiful book by Eric Kandel. It's called something like Reductionism in Neuroscience and Art in the 20th Century. And he goes one by one through all the modernist movements from New York and tells you what they were doing innovative from the point of view of neuroscience. And the funny thing about Pollock is that it hijacks the mechanisms of detecting movements to guide your attention. So when you look at the Pollock painting, it is moving you here and there. You cannot stay quiet, stay put in front of a Pollock. It guides your attention to the important features, which there aren't any important features in the Pollock. It's just exploiting the mechanism. I wanted something like that, something that guides my attention to the things that are important when a network is given. And somehow, when you do something like clustering, you come up with communities in a network. And this is somehow you get some automated analysis that tells you where you have more density of links within this cluster than from this cluster to the outside. This is a little bit really like I don't know anything about this network, but automatically I can be guided to this outstanding community, this outstanding community. Something that also didn't satisfy me is that this kind of analysis is going to guide you to communities, to outstanding features that are close in space. If there is something important about the series of nodes that makes them similar, but this does not depend on the fact that they are close in space, then something like community detection is not going to be able to detect it. Imagine that you have a series of communities. Imagine that these were perfect communities, all right, and that there were just a few bridges from one to the other. The bridges connecting this and this are not going to be connected to the bridges connecting this and that, but the role of the bridges is similar, disregarding the fact that they are not geometrically close within the network, all right? So that was the whole motivation, to look for something that's going to automatically guide my attention towards those features of the network which are important. And somehow, if we can transcend geometric closeness between the nodes, if we can go somewhere that tells you, well, you know, these nodes here, they play a similar role, even though they are not even close in space, right? That was the whole setup. So I went to what I think is the simplest thing that we can do. First of all, we get the network and we measure a lot of things, as many as we can imagine. In all the examples that I'm going to show, we have this 19 different things that I measure. There are things related to centrality, like all of these. You can think that they are very similar. I will show you that there are nuances to that. There are things related to how central nodes are in a layer by layer setup of the network. There are things related to cycles. There are things related to average path lengths, clustering, so typical suspects that you can measure on a network. So just go and measure everything that you can measure, all right? You might already get a hint what I'm going to do next, but before doing that, I was also interested not only in the properties that a node has, but also in the properties in the neighborhood of a node, all right? So for every node, apart from these properties that I call primary properties, I'm going to measure the average of those same properties in the neighborhood. This is going to tell us things such as, is my network assortative? Like, if a node has high degree, is it also connected to others with high degree? Or we can extend this to other properties. If it has long cycles, is it connected to other nodes with also long cycles? Stuff like that. And also I measure the standard deviation of each property over the neighbors. Why? Because if this standard deviation is very narrow, this is telling us that this node is selective. I only connect with those that have this very specific property. If the standard deviation is broad, then this means that my node is very promiscuous. It has no preference to connect to this or that property-wise. So what we are left is, for every node, a huge list of properties. Every node, a huge list of properties. And probably at this time we already know what to do with this. We apply some, every node is going to occupy a point in a huge dimensional space. We cannot visualize this. So we do some dimensionality reduction to be able to work with these kind of things. And the simplest thing that we can do is a principal component analysis. I know this is probably the oldest technique of this kind. There are others which are newer, but let's stick to the simplest thing. And then in the future, we can elaborate and do more papers with more complicated techniques. All right, so from properties for every node, we move to correlation matrix. We move to some eigenvectors, and this simplifies all this structure that I just told you. Let me go to a specific example, simplest one that we can imagine. It's a Watt-Strogatz network, okay? Imagine that you have a Watt-Strogatz network before doing any rewiring, okay? In this case, every node is going to be connected to the next neighbor and the next neighbor, all right? So you're going to have a ring. Now, if you measure anything on any node on this network, all the nodes are going to have exactly the same properties. They are going to have exactly the same number of neighbors, exactly the same clustering, the same number of cycles, same length of cycles. This is going to be very, very boring. So if we measure all these kinds of things, when we come here, we are going to have a list of numbers which are all the same. This is just one point in a very, very high dimensional space, all right? Now we do the Bhatt-Strogat sin, which is with certain probability. We break a link and we make a shortcut. And when we do this, we put a defect into the network. We remove a triangle. We make a connection which is far away. This changes the average path length of the node that has been rewired to the rest of the network, and also of the nodes nearby. This changes average path length, this changes the length of typical cycles, this changes clustering, changes between the centrality, and this kind of thing. So when you introduce one such a defect, you alter a little bit those numbers that we measured for every node. And all those numbers, all these nodes that were represented by a single point in a high dimensional space, they start to diversify, and they start looking like this. So what we have done here is we measure all those properties, we projected them back into three-dimensional space to be able to visualize them. If all the nodes, if there were no rewiring, they will all be living here, but since there are defects, they start moving around. This is color-coded. I'm sorry for the colorblind people. This is the best way I came up to do representations. This is red, this is green, this is blue. So something very reddish means it is located very forward in the first principal dimension. Something very green is very forward in the second one. Something very blue is very high in the third one. And what we can see here is that this color and the position in this space encodes somehow a sense of topological similarity between the nodes. All the nodes that will be similar to the original network without rewiring will live here. They will all have the same properties. And when we go to the network, we see that all the nodes that live in a fairly regular stretches of the network that looks like a grid, they all have the same color. They all live in the same place because they are topologically very, very similar to each other. All those nodes which are near some defects, for example, these ones are at the center of the network, they appear very, very green, very likely because they have higher centrality values such as eigenvector centrality and between the centrality. I'm not so sure about between the centrality, but certainly they have something different. They are topologically different to those over here. Now we can go back to the original network that motivated all this work. This is the same analysis performed on the collaboration network of the Spanish National Center for Biotechnology. Once again, every person is a researcher from that institution. Two researchers are connected if they have co-authored a paper together. We measure all those things on all those nodes. They live in a high-dimensional space. To be able to visualize it, we go down to 3 dimensions. We only do this for visualization. Every analysis that follows takes into account all the dimensions. It doesn't really make a difference. Three dimensions are usually very informative. So once again, this is color-coded. Red means higher in PC1, green means higher in PC2, blue means higher in PC3. And we see that the distribution of nodes in this space is different to this one here. The PCs are different. They are not the same. Each network is going to induce a different relationship between the properties. I will say more about this later. And we can see some structure here. We see some nodes with live up here, others with live up here. If I was able to rotate this, I should bring up something that allows rotation for the next presentation. But there's actually like a depression here, and this cloud stands higher than this little thing here. So this heterogeneity over this principal component space is capturing the heterogeneity of topological properties of the nodes of this network. And we can see somehow that this bluish thing is going to correlate with something very central to the network, right? So we can use this to define communities. And the communities that were defined before that I gave as example, they are classic communities from clustering methods. And I termed those geometric communities because they are based on the fact that nodes are geometrically close in the network, geometrically understanding the distance that the network induces. Now I'm going to term these communities topological communities because they are based on the distance between topological properties of the different nodes. So we do some hierarchical clustering in which starting from this space, we start grouping up together nodes which are closest, and we find the centroid, and then we keep grouping them up until we have everything together. And if we go backwards, if we start from the whole network and start splitting in those places that are going to separate the network better, we have a hierarchy of communities that arise. The first thing that happens is that the network is split in two. This thing in red, I'm going to call the core. It stands pretty much in the center of the network. The rest in black, I'm going to call the rest of the network. We can go to the next level. It splits again. Now, the rest of the network splits into something that in this case, I call a shell that separates the core from the periphery. We can go back another level, and we see that the periphery splits in two. There are two kinds of periphery. In yellow, there are the leaves of the tree. Those are researchers from this research collaboration network who only had one collaborator. They were probably either researchers who don't interact with anybody or people who just arrived within this period. They didn't have time to make too many papers. Then you have another kind of periphery, which is in blue, in which you have, they're probably small groups. They do have a fairly amount of collaborations, 4 or 5 people, something like that. They are peripheral somehow. Interestingly, neither of these two communities is contiguous within the network. You cannot move from one to the other unless you visit another community in between. This means that there is no algorithm for community detection, from classic community detection, that's going to spot out this topological difference, and that's going to tell you, well, you know, all these yellow people, they are kind of the same. All these blue people are kind of the same. If we go down another level, the core splits into two, something that I call the core again, and a reach club. And a reach club is a department in which basically everybody writes every paper together, and they don't collaborate much with anyone else. And because they write so many papers, they are very, very central to the network. And also interestingly, we see that to separate the different communities, we need, so for example, to separate the reach club, we need to split along the third principal component. To split the core and the shell and the peripheries, we will split along the first component, which is correlated with measurements of centrality. And to split the two different peripheries, we need to split along the second component. So this is what I was looking for. This is an automated way to guide you to the things which are important and to guide you to those things in order. So now let's turn to this kind of analysis to something more meaningful. Nobody really cares about collaboration network of a Spanish research center, even though it's a pretty big research center. So let's go to something that people care most about. I'm going to split it down. I missed it. It's not super important. I already say everything that we needed to say. So let's go to this new network here. This is a new network. I'm going to tell you right away what it is, but just looking at the same analysis, every two nodes are connected if whatever happens. And you can see, so we measure everything that we can measure about every node that we have in the network. This lives in a high dimensional space. For visualization, we project this down to 3 dimensions. And here we see some structure that looks different to the 2 ones that we had before, because each network induces its own shape on this space. This is the global transport network. So every node here is an airport, and 2 airports are connected. If there is at least 1 flight, these are the 500 most busy airports as of the time when I took the data from. This has changed because now the airports in Berlin have been substituted by a single big airport, which is actually important to understand something that is going to happen next. So just looking at here, again, we guess that there is some structure. When we project this into the network, we can see pink things which are not necessarily connected. And once again, this is telling us that this analysis is detecting features, it's detecting topological classes that don't depend on the fact that 2 nodes are connected to each other. They might be far away and still play a similar role within the network. If we apply the composition into topological communities, as I did before with this hierarchy that unfolds, we have some very interesting insights. For example, this black community here, it encompasses most of the airports in the United States, except for the very, very big airports. And this is super interesting because this is the only topological community that it's also a geometric and geographic community. It happens that in the United States, all the airports, except for the very big ones, seem to be more similar to themselves topologically than to any other nodes in the network. And this is probably so because the US invested a lot into having air transportation, making this the main thing in the United States, as opposed to Europe, where we have a lot of trains and otherwise. Then we have the red topological community, which is what I call the global transportation backbone. This is the network that holds the whole thing together. This is an international network with the main airports, including those in the United States. And it's very interesting that you can say that those in the United States integrate better in this global transport network. And then we have others. These are two airports in Taiwan, so probably an oddball. And probably the smallest airports that made it into the top 500. And the other two are mainly defined by their more or less peripheral role. We get closer, we can see some funny things. For example, some interesting Mexican airports are parts of the United States topological community because they are touristic hubs somehow. Also, in Europe, we can appreciate all the airports that belong to the main global backbone, and we can see that there is only Prague belongs to the global network from the former Soviet bloc. Berlin did not when I did the analysis because there were 3 airports. Probably Berlin now is integrated in this. I need to redo the analysis with the new data. And also you find some such big airports from the global hub in Southeast Asia, but not in Africa, not in Latin America, interestingly. This is what I took the center of mass of each one of these groups, and I got sorry. These are the different networks by themselves. Sorry, I had to do representations. This is the last one. So this is what the United States topological community looks like. This is what the global backbone looks like. They seem more similar, but this is better integrated than this. Actually, when we move to the next topological community, we can see that there are differences in topology already visible. And then that's what everything was about. By the way, this one is only the largest contiguous components of the green airports, because it's also non-contiguous, meaning that no classic community detection algorithm will be able to detect that all of these are topologically similar and that they should be grouped together. And this we can see here in this slide. To do this, I just used a classical community detection. I don't remember what it was precisely, but probably one of those maximize intergroup links and minimize groups to the outside. And what we can see here is that in this case, communities are geographically put together, but it cannot tell apart that the fact that there is a distinct topological group that connects the whole network together, that is not localized, that spans some airports in the United States, Europe, and so on and so forth. Right? So if you're given a new network and you don't know what to do with it, I think you should do these 2 things. One of them is, well, look me for classic communities, what I call geometric communities. Tell me where they are, and this has already been done a lot of times, and I think it's super informative. But I think that this is missing out second part of the story, which is, okay, find me nodes which are topologically similar to each other, even though they are not near in space, or even though they might not be connected within the network. Some other examples. This is from Brain, from Connectome. We have a large database that we are still analyzing. I'm going to give you just 3 examples with some insights, which might vary when we analyze the rest of the 1,000 something brains that we have. Now, again, same thing. We have all these nodes. Every node represents a little piece of cortical matter. In this case, 2 nodes are connected if there is at least one axon that starts in here and ends in there. So it's an anatomical network. We measure everything that we can measure network-wise for every node, and we project this into 3PC. And once again, we see that this looks different to the clouds that we had before. Why? Because every network is going to induce its own shape into this space. It's the shape, the cloud that we have here is going to be characteristic of the network. 3 minutes? Okay, then I'm very, very bad with time. Okay, so let me just go very briefly through this. What we find is that there is interesting. Okay, there is. What we find typically, and here I'm just showing the most typical brain, what we find is that there is a first topological community that groups together nodes from the visual area with nodes of the somatosensory area. And this we have observed in many brains, but I haven't done a proper statistical analysis to prove that this is robust. But this will be telling you that topologically, the visual system and the somatosensory motor systems are more similar than the rest of the network. This is an example to say that some brains are very asymmetric. Usually, nodes in the left are topologically similar to the counterpart nodes in the right, but some brains find that there is more topological similarity within a hemisphere than for every node to its counterpart. I will just gloss through this because I wanted to come to, I wanted to show this. So what do you measure when you measure something in a network? When you measure clustering, when you say, okay, we have a page rank centrality and we have eigenvector and we have harmonic centrality and so on and so forth. Are these things similar? And the answer is, this depends on the network. Not only a network has a relationship between the nodes and has a classification of nodes depending on their topological properties, but also the network induces similarities and dissimilarities between the different properties that you might be measuring. Something that happens often is that the first component aligns with centrality. A lot of measurements that capture centrality define usually the first component, and you find those measuring centrality and some that counter-align with centrality, such as clustering, which also has an interesting story behind it. But this is not always true. For example, when you go to the Watzke-Strogatz network, everything falls apart. And centrality is usually not, so in here I say I contrapose centrality with, I confront centrality with clustering, but actually in the Watzke-Strogatz, the eigenvector centrality is well aligned with clustering. So what I mean to say with this is that what something is measuring also depends a lot on the network. I have to finish now. I had more things that I wanted to say. Just some examples from this is a network based on programming languages. Two programming languages are connected if one was inspired by the other. This is a directed network, but I collapsed it into an undirected one. This is once again different. There is a core, and there are two different of peripheries, but there are two different kinds of peripheries that we find in other networks. This analysis was able to retrace the splits in the United States Congress and Senate. But I will not get into that. And I wanted to come here. This is a recent paper. This is final slide, almost final. So this is a paper from a couple of years ago in which they have a network. They reconstruct the network of interactions between proteins in the gist. This is the largest network that I was able to analyze. I don't have the full analysis here. I just wanted to put it here in case there is someone in the audience who is an expert in this kind of things, because I would love to go deep into that. You can see very, very rich structure from the analysis in here. And I'm very happy because this was the largest one. Why? There is an issue. This method, unfortunately, doesn't scale very well. One of the things is because you have so many nodes that things such as between the centrality, they take forever to calculate. But then the other thing is that if you have a heavy tail network, then whatever is in the heavy tail, the degree distribution, the degree of the nodes, is going to bias everything. That's because if you have a heavy tail network, because you're going to have a variation over many orders of magnitude, that's going to be your first principal component. And for visualization, that's going to be ugly because it's going to stretch the whole space. So basically, the first dimension swallows all the information that you can have from the others. And probably I have to modify that using logarithms, stuff like that. Anyway, this one is a happy network because it hasn't got a power law distribution, so it leads to this very nice representation. And I think this is it. Thank you very much.
Okay, thank you very much for a very nice talk. So we have time for one question.
Any hope for doing any of this for signed networks? Any hope for doing any of this for signed networks? A network that is a sign.
Oh, something important
Sorry, something important that I didn't say is that all the analyses I perform are for undirected, unweighted networks, but I think that you can apply this to anything, going from this to signed networks, to weighted networks, directed ones, to simplicials, to so I hope that this is the beginning of something that we apply. It's like you're given a signed network, you don't know what to do with this. What do? I measure those things, and I let the network tell me what it is important for her, somehow. So yes, I hope. I haven't done it yet because I didn't have time or hands.
So one very, very quick question.
That's one simple question. So are you observing very different main differences between the networks? For example, the protein-protein interaction network will be full of noise because we don't know, and I don't know. There are techniques like this to hybrid, but they are very noisy. But on the other hand, you have the global transport airport that will be very precise. So you are observing that are very different. Very different. You are observing the noise clearly, definitely. That you see, this is very dirty, or you are observing fundamental properties in the.
By now, I go one by one. So I've been doing this for the last 2 years, and I haven't had anyone to collaborate with or to expand, and I don't have really. I need more hands and more time. I think that there might be some universal stuff, not universal, but some patterns that you can recognize. But so far, I'm going one by one. I look at the network and I see what it tells me. So something that I try to do, for example, with a master student, with the brain networks, for which we have 1064, we wanted to see how the PCA from one brain relate to the one of the other, to see if there is some consistency, to see how those asymmetric brains arise, how they rotate the principal components and this kind of thing. But we didn't go very far in this analysis yet, for a matter of time mostly.
Okay, so let's send the speaker again.
Thank you.
Our next speaker is Professor Christian Maes, who's going to talk about non-equilibrium-induced kinetic selection, pattern recognition.
So, thank you very much to the organizers, especially to Joachim, for helping me so much to get here, and I'm very happy to be here. For the local people in Granada, it's really a pleasure to see you back. The title of my talk is related to something which is on the, I believe, work in progress on all of molecular statistical mechanics, having to do with what we could abbreviate as kinetic selection. There are like two models to that short talk. One is that despite many ways of speaking about dissipation, I think we should remember that dissipation is also an opportunity in the sense that it will be, many things are thanks to dissipation. There will be often that word on my slides, and maybe towards the end I will tell you why that is a word, but basically flying of birds has been a paradox for many years, since the D'Alembert paradox in the 18th century, which proved that birds cannot fly if you apply just Newtonian mechanics. It has to be understood really that it is thanks to dissipation that birds can fly, and I think that this idea of thanks to dissipation is something that we should cherish a little bit more when doing problems. The second model is that it has been touched already, I believe, this morning, that despite all the great things and the attractivity of machine learning and all of that deep learning, there are also some disadvantages, maybe I express myself, and that is that it often comes a bit as a black box, and one could say that it leaves a bit the traditional scientific attitude of trying to find for mechanisms. And somehow when it comes to the inspiration of statistical physics, it uses often a minimization process of minimizing a cost function, like minimizing energies, which is in fact not at all, I believe, how life is working. And therefore we really need also in a little bit in the spirit of previous talks, we really need to come to terms with how to do statistical mechanics of trajectories. Okay, so the main message indeed is dissipation is not just losses, but is something which we don't need to avoid really, and interesting physics, science comes out by these ideas in terms of selection, steering, control, and so on. Okay, so this is a heterogeneous audience, I understand. Not necessarily everybody is working daily with thermodynamics or statistical physics, so allow me to give you a little bit of an introduction just to make sure we understand what we're talking about. So the first encounter with dissipation, well, has to do somehow with the history of statistical mechanics. The first in statistical mechanics started with the statistical interpretation of temperature, namely as kinetic temperature, somehow related to kinetic energy, and the nature of heat in the first law. That was followed by these wonderful studies of Sadi-Carnot, Réflexions sur la puissance motrice du feu, where there was great progress, great applied science, which evolved from a very abstract understanding, conceptual understanding of what a heat engine was, and mathematics entered at the same time, treating these theories of heat and dissipation. Now, that story of Carnot often has been concentrating on the idea of efficiency, and that was worked out then later by Kelvin and Clausius. And one of the great things that happened was that one identified a state function. That is the Clausius relation, namely if you have a quasi-static reversible process and you look at the heat divided by temperature, it is a state function, and that defines the entropy. And then one could really speak about dissipation in terms of entropy increase and entropy production. That's more or less the short history of how that worked. Dissipation entered thermodynamics. But then something very strange happened. Something miraculous happened, namely Boltzmann showed after some attempts that that same Clausius entropy in equilibrium can be identified with a measure of degeneracy, namely the logarithm of the ways you can make a macroscopic condition given constraints of, say, particle number and energy. So in other words, it gives you a volume in phase space collecting all the microscopic states which realize a particular macroscopic condition. A priori, I mean, it is not understandable just by looking at the definition that that has anything to do with dissipation or anything to do with heat of Clausius, but it does. And in fact, there are other miracles that happen. Not only is the same entropy speaking about heat and dissipation, it also speaks indeed about fluctuations, as in the Boltzmann formula. It also relates to the arrow of time. That's another aspect of that entropy increase then and dissipation. But it also governs linear response. Think about Kubo and Green-Kubo relation. And it also gives you the idea of statistical forces. The gradients of the entropy gives you the statistical force. So in equilibrium, or around equilibrium, there is only one entropy, and it does all that job for you. Now, if you go to non-equilibrium, there is no reason in the world that one particular object, like the entropy or the entropy production for that kind, would do all that together. So in non-equilibrium, over many, many years, there have been this obsession, I call it an obsession, with entropy and entropy production, and trying to find what is somehow the extension of the variational formula, what is the extension of the Gibbs-Boltzmann distribution. These kind of obsessions have not really done a great deal. What we really have to do is to really allow for, well, non-thermodynamic information to enter our studies. And that is in particular true whenever we are doing non-equilibrium processes. This is still another example, a quote of Boltzmann, who is speaking about, living creatures, somehow a reference to biology, and he is emphasizing it is a struggle for entropy. You have to survive, it's a struggle for entropy, but that is what somehow in these days was the main thing to look at for understanding the secrets of life and things like that. Okay, so now just to make sure, after my introduction to thermodynamics, let's see that we understand what we mean by non-equilibrium, because often people think that non-equilibrium is something that is relaxing towards equilibrium. That's not what we mean by non-equilibrium. I mean, with no offense, you are all kind of non-equilibria persons, I hope, for a long time still, because you're full of energy that is pumped around. You have currents, steady currents, at least on certain time scales and energy scales, you're very much stable and stationary, but you're non-equilibrium. So other examples you can see there, you know, the metal bar between two temperatures, say these outside and inside of a cell which are connected by an iron channel, but also look at Escher here, where you have this strange kind of, you know, these litos with the staircases, that when you go up, you really go back to yourself. So that's full of non-conservative effects, non-conservative forces, rotational forces, and so on. So that's steady non-equilibrium, and that is what we are really talking about when we are going to stress the importance of dissipation in a kind of paradoxical way. So that is non-equilibrium. We speak about steady non-equilibrium, where currents are maintained, they are non-vanishing thermodynamic forces, and all the time there is Joule heating, there is all the time dissipating power to the environment, and the time reversal symmetry is broken. That's the summary of what non-equilibrium is. And I hope you take yourself as a good example. Okay, now what I wanted to speak in this talk, shortly though, and in a very simplistic way, is the good news that dissipation has. Nothing to avoid really. Namely, it is possible, thanks to dissipation, to do selection, evolution, steering, controlling, and to have emergence of all kinds of different phenomena which really are close to our heart if we are interested in things like physics of life, new materials, or other aspects of where complex systems are related. So I think that these ideas, which are conceptual of a certain type and that I will illustrate with very simple examples, are somehow important for you. Here they come. Okay, I have another motivation, maybe I should skip that, but just it's also about us that so the traditional approach to learning, you know, associative memory has often been, it is variational. You do steepest descent with some cost function, think about the usual traditional models, but I cannot imagine that my brain is doing that, and I hope it doesn't. That there is not such a minimization of energy and that I somehow a picture in my brain and not getting out of that local minima for a long time. I mean, that would be a disease, not something that I would like to have. I mean, this is good for artificial intelligence, but I want natural intelligence. Okay. By the way, we more or less know that nature, you would like to understand natural learning procedures, but we also know that biology and the brain is producing heat, so it is really non-equilibrium systems. Okay, so heating does not necessarily mean losing. We need to be a little bit, we dare to enter this kind of dissipative physics. Okay, now I'm not going to tell you that the brain produces heat, probably you know that. Okay, so let me go to the simple example immediately. So this is actually not my example, this is already from 1975, this is called the Voltor's theorem, because in that time there were various people that started to believe that they, you know, I was telling you about the obsession with entropy production. People are speaking about how you have to do Maxent or not Maxent, you have to do minimum entropy production principle. The others say, oh, it's maximum entropy production principle. And so Landau wrote a paper that none of this makes any sense. In fact, it's wrong if you test it with examples. And the idea, I will just, this is not the example that he did, but you can as well give that example. Think about a jump process. So it's like a reaction process. It's not like a diffusion, and we think simply, I believe, about a ring of vertices. These represent the states. No, it's abstract. So the rings are, the sides are called x, and then clockwise you have the x plus 1. And there are transition rates. So think about a walker that will jump to the nearest neighbor on the ring. And in blue there, you have the rate to so this is the rate to go back or counterclockwise. This is there's a minus. You see there's a minus in the exponent, so it's biased to go forward, biased to go clockwise. This is the epsilon here, which is like the electric field of the external field that drives the motor to do clockwise. But notice that I have here over that link a particular number AX. So I just call it AX. It's really between the link X and X plus 1, which is the same in the blue and in the green. So this is what we call time symmetric. Now, if you go backward or forward, you have to meet the thickness of that link. So now, what this is doing? Well, in equilibrium, it is doing nothing. What is equilibrium here? By the way, equilibrium is that you do not have a bias. If epsilon equals to 0, then you don't have this rotational force. If you're in equilibrium and you just have these AX, whatever they are, as long as they are not zero, all the states are equally probable. There is no selection. But now suppose we are, thanks to the epsilon non-zero, thanks to the fact that we dissipate and we have Joule heating, thanks to the fact that epsilon is non-zero, let us now look what is the state which is selected. But in fact, the AX now start to be enormously important. This is the blowtorch of our model. Where you can have AX which are very big here, and here maybe they are very small. Very little. So it's kind of extremely easy to understand. And what we're getting is that the stationary distribution, so where you spend your time mostly, is inverse proportional to these AXs. But I mean, even though this is a trivial example and a trivial result, it is fascinating. I tell you why. Because it means that if I or nature is able to direct these AX, from the moment you have a good enough epsilon, this is, by the way, a second order effect, so life starts in second order, I don't think in first order, then you see that you can select the state by the AX. If you take an AX to be small somewhere, you will have a high probability of this rho X, right? So you can select the stationary distribution. So this is an example here with just a very simple example. Again, you have now a tree state. I just give you here the rates. I hope you can see associated to the arrows there are rates. Again, it's clockwise that you have a bias, counterclockwise there is no bias. The epsilon is the thermodynamic force, so to speak. If you just look at the, I got this computer. You look at the stationary distribution, you will find that you can get a lot of differentiation depending on how, for a particular epsilon, you are arranging it. So away from equilibrium and to second order, starting in second order, from equilibrium, you can select. It's great. Okay, so that's an example of what I call kinetic selection. It's kinetics because the AX is not part of our thermodynamics. It's more like a time scale. It's a frequency. It's also what I refer to in more general discussions to a frenetic contribution. Okay, this can be used actually for giving models of frenetic memorization or classification, but I believe I do not have quite the time to do that, but this is one of the works we did where you start from a complete graph and you have patterns which you select there. Maybe here it's just the 2 and the 1 which are patterns. This is the cat and the dog type scenario. And now you randomly have, you start with a random orientation of all the links between the nodes in the complete graph. And what you are doing then is that you basically make loops so that the cat and the dogs appear in a different loop. Then by doing this frenetic steering, by controlling these AX, you can let the system evolve towards the cat or the dog. So without this idea of falling in potential well, and then you drop there of the field. Alternative way of doing that. Of course, very simplistic scenario. Okay, so this is what we call frenetic steering. I think I have said enough of that. Some kind of a more well-known from around the same period as the blowtorch theorem was the, of course, the Nobel Prize winning ideas of Hopf and also there in the which is a similar
similar thing, namely you have to make a distribution which is not a good distribution. Otherwise you're dead. Right? So this is the error correction it is called, which I think kinetic proofreading is maybe a little bit better, where you can increase certain production expressions not really available if you just look at the energy landscape. So it is time somehow to start thinking about other things than landscapes. Landscapes are great, it's like reducing thermodynamics to mechanics because you can compound and things like that. But there is much more to non-equilibrium than landscaping. Okay, so I'm skipping other examples. Another thing is that here we have been speaking about the selection of states, but you can also select currents. So it can happen that you have a network, and this is just representation, where the blue direction is counterclockwise, the red direction is, the blue direction is clockwise, the red direction is counterclockwise, but if you look at the details of this network, they produce the same entropy. So they have the same entropy production. So then the question is, what will happen? What current will be selected really? And the answer is the thing that will be selected is again kinetically selected. That is basically also related to the motion and the direction of motion of molecular motors. It's based on the same conceptual ideas. It's not something like a thermodynamic force is pushing in one way and not in the other. No, it's kinetic. Okay, now let me finish, I think, that talk here. So by describing two classes still of control, which are also a bit on a simplistic conceptual level, but just to give a bit of understanding of how you can control the equilibrium thanks to dissipation. So the first class is what I mean by you control the mean force. So let us see here. So think about the fact always here that we are coupling one system to another system. So here is, you think about the slow system, you have a probe that can move on a circle. And it is coupled to a chemical reaction with three states. This is a non-equilibrium, that's the green thing here, it's non-equilibrium, it's a biased thing, and they are coupled. How are they coupled? I'm not telling you the details of how they are coupled, but there is a joint energy, U, X, Y, where X is where the probe is on the circle, and Y is the state of the chemical reaction. So there is a coupling. Now, what is Newton telling us? There is a force which is minus nabla this energy, where you take a derivative with respect to X. But now suppose that this is much faster, that this chemistry is much faster than this more macroscopic motion, then I can average over the stationary distribution of the chemistry given a certain probe position. So now the probe has what has the time to move and the chemistry already done at that particular probe position. But this is called the mean force. Now, clearly, you need to, if you know, what is now the point? I want to influence that force, that's steering, that's control, that's optimization. I want to control that force to see what is happening to that probe. I want to take a hold of it. But what does it mean? If the U is given, if the landscape is given, there is another important thing still, and it is these brackets. Because these brackets are the stationary distribution of the chemistry. And that's a non-equilibrium system. And that can be selected, as we just saw. That was the frenetic steering. That was this AX in the simplest example. So you can use the kinetics, the frenetic, the time symmetric component. So that's the paradox somehow. You know, you're in a world where the time symmetry is broken, but it turns out that because of the fact that it is broken, because of the fact that there is dissipation, the time symmetric fluctuation sector becomes important. It gets ignited, so to say. And it is that selects possibly the stationary distribution. In other words, that controls the force. And it is this force which will no longer be gradient. It could be a rotational force. So in other words, you will have that possibly this green one will move in this direction or in the other direction. But you will get a current by selecting a particular stationary distribution, which you can also change in time, by the way, by slowly changing the chemistry parameters, which always relax at different ones. So this is a first class of examples. Maybe you don't want me to do the mathematics and all that, but I think you got the idea by my showing it. Okay, so this is a not biological example. This is about the weather. Somehow, you know, the weather prediction. I not speak about that too much, but this is just a Lorentz model, which is a dynamical system. You can simulate it in that way. So instead of one probe on the circle, you just go two-dimensional. You're three-dimensional. And what is guiding it, so now you're sitting in the car of the Lorentz, and you're guided by the chemistry, which is telling you where to go, basically, via this mean force. And in this way, you can simulate, for example, the Lorentz model. Just a pleasant example, nothing more. But I think I want to, okay, so there are many more examples of that kind. You know, you can imagine that once you can control this mean force, you can also stabilize patterns, which were unstable in equilibrium. So if you just put probes in a circle, in a toroidal trap, where the viscous fluid, which are these green colloids there, they are driven, they are red arrows. You're in a non-equilibrium medium, and you have many probes now, which are the gratings. These are the probes, these gray ping pong balls. The non-equilibrium driving is done on the medium, which is these green things. They're pushed around the ring. You can show that there arises a non-reciprocal interaction between these probes, which is repulsive, and which is such that you get a crystal pattern, which is only there in second order around equilibrium. Only if the colloids are sufficiently driven, thanks to their dissipation, you can stabilize a crystal pattern, and you can make bone states and all of these things, these kind of toy examples. Just as manifestations of this general slogan that kinetics matters, that the frenetic component can be used to steer, to select, to control, and to stabilize certain patterns. Okay, let me move to class number 2 to end. So I'm not going to discuss the flow towards this crystal formation, nor how you can balance a ball on top of a Mexican hat. But let me go to the second class. Okay, these are some references for the class 1 of frenetic steering. But let me go to class 2, which is of a bit of a different kind. Instead of controlling the mean force, you can also control the violation of the Einstein relation. So you know that if you are in equilibrium, I didn't explain that, but if you have an equilibrium environment, there is a general way in which noise and friction are related. That's called the fluctuation dissipation relation, or Einstein relation. So it has been a lot of papers have been written saying FDR is violated, Einstein relation is violated, you know, there and there and there in every system. Of course it is violated, because we are alive. Luckily it's violated. The question is, how is it violated? The question is, can you do something about this violation in a control way? In other words, can you somehow make a friction that you want? Because if it is violated, it gives you freedom, right? It gives you freedom to control. So here is just this class 2 is control motion via a particular violation of fluctuation dissipation. So this is a quick reminder. I'm sorry for those. This is Wikipedia movie of the Brownian particle and an equilibrium medium. And then we all learn in school that the effective noise correlation in time is related to the friction. It is called Einstein relation. Okay, but now if you are going to non-equilibrium medium, so you have this ping pong ball, and then you have here active particles or somehow chemical reactions around you happening which are coupled to you, different time scales, you wonder, well, the Einstein relation will be broken. Also when you have active systems, skipping all that, and you get, for example, pictures like this one here to the left. I'm not sure. Here you see these red particles are in non-equilibrium. They are in fact active and a tumble or active Brownian motion, and the probe is going to get an initial acceleration actually. So it's like in this painting of in the Sistine Chapel where you got this. So like the active medium gives activity to the probe. So the probe becomes an active particle. It was dead in the beginning and it's
deselection, in the sense that it starts to have an active motion, so it accelerates and starts to become, that it saturates at certain velocity which are compatible with the speed of the run and tumble particles. So you give activity from the medium to the probe in that way. A very simple mechanism, but that's by controlling this kind of second order fluctuation dissipation relation. Okay, let's see. So the friction, indeed, you must be able to make something which is negative. So for those who know that there are various parameters in activity, like resistance, if you have high enough persistence but low enough propulsion speed, you are able to have the inheritance of activity from the medium to the probe. And it starts by creating negative friction. So in the vertical axis is basically the drag coefficient. And you see that there is a regime of negativity if you have sufficiently good life in your medium. And you can control it via these parameters to understand what you get. And that is the kind of run and tumble process you see if you look at the trajectories, that it really starts. This is the probe motion, probe trajectory becomes active. It's no longer a Maxwellian, it's like a double-peaked Maxwellian, automatically. Okay, I think I will stop here and not trying to extend any other things. I think that this really good, okay, let me not skip the history. Maybe just this quote again, this was quoted by Longower, a quote by Charlie Bennett, which I will read to you. So it said that, suppose I have a set of planetary conditions, and you want to understand whether life is a preferred state or only something like transient. Then he says, it's not just comparing the life as a state and death as a state. It's about escape rates, it's about accessibility, it's about lifetime. These things are kinetics. And the good news is that we start to learn about this zoology of kinetics, of all these different aspects of this complicated dynamics, how we can see what is important of them. What is the way of speaking of them? These theoretically and in simple models, and this is what we collect under this name of frenzy, which is really somehow the complement to the entropic contribution in the understanding of this physics. Okay, so let me start. So here is what flower is alive and what flower is dead? It's not a here the story is that when Western civilization has been thought about Hamlet's question, are you alive or are you dead? It just takes snapshots of these doses and you see which one is alive. But then, on the other hand, if you look to the trajectory, you start to see which one is alive. But only on the level of trajectory, not on the level of trajectory.
Okay, so thank you for a very inspiring talk. So we have time for one or two quick questions. Okay, so if entropy makes everything in equilibrium, which is your favorite set of observables for non-equilibrium frenzy?
Yes, certainly the dynamical activity is important, which is a time symmetric measure of the number of transitions per unit of time. Another thing is accessibility and escape rates. These are, if you look at the simplest processes, which on the level of chemical reactions or jump processes, if you look at the dynamical fluctuation theory, then you have one part which is entropic, which is related to dissipation, and the other part is exactly related to this dynamical activity and escape rates. So to start, these are really the things to have in mind. And that these control whether something interesting happens.
One more question.
So then the conclusion is, whereas we are moving, we will be alive.
I'm happy to hear that conclusion. I was not sure it was my conclusion, but let it be.
So then, even after death, we still are alive. because our molecules.
Let us get the logic clear. What I told you is life from life. I didn't say you cannot die. But if you won't have life, you better have life around you.
Okay, thank you.
Okay, so let's thank the speaker again. Okay, so the last speaker of this morning session is Professor Sebastiano Skorsak, who is going to talk about a stochastic multi-scale framework for chromatin folding.
What is course? Course. Can you write? Yes, yes, this is it. Yes. Oh, just open, no? Okay.
So my name is Sebastian Korsak. Actually, I'm not a professor. I'm finishing my PhD right now. I'm making a PhD in chromatin, in the 3D structure of chromatin. And in general, this work is work about how we can derive, let's say, a physical model from some biological observations, which is not always very easy. In general, chromatin is a very high-dimensional being, a very dynamical being. Some people say often that we are not ready to model it maybe physically if we already don't know like the function of proteins very well. So let's start. First of all, what is protein? What is chromatin is DNA with proteins. It's a very dynamical thing. It involves a lot of non-equilibrium processes. Why we simulate chromatin? It is because in chromatin actually we don't know what is the 3D structure. We don't have any crystallographic experiment or anything else that it can provide us the exact structure. So we want like to create some virtual eyes, let's say, to see the structure somehow. And in this work in general, we focus on the whole cell cycle, which means that we don't care only just for a static structure, but we want to see how the structure changes during DNA replication. Okay? And of course, we also care about the condition of cancer. So this is the reason why I tried also to model replication stress. So let's move on. So these are the scales of chromatin. We start from the finest scale, which are nucleosomes. This is the packing unit of chromatin. Then we have chromatin loops. In this situation, we have two main protein players that interplay. It is SMC complexes and CTCF. SMC complexes are highly dynamical. They bind and unbind in the DNA and they extrude loops, while CTCF complexes are boundary elements and they are very stable and they are some kind of they indicate actually when the loop extrusion will stop. Then in the higher scales, in 3 megabases, we have A and B compartments. And in this situation, we don't totally understand what are these A and B compartments. We know that they come, let's say, from maybe from some histone modifications, maybe for something that happens in general in lower scales, but it is probably a collective effect, and we see like large areas of chromatin, let's say, to cluster together. Yes, and it ends, they also have some biological meaning that B is closer to the lamina and it is more condensed, whereas A, it is less condensed and it is more active in general. And then of course we have chromosomes, yes. So this is the spatial complexity, but we also have dynamical complexity. And this is even more difficult because this is where non-equilibrium comes. And so, for example, we have like the G1 phase. This is, let's say, phase where we have mostly loop extrusion, a stochastic effect. Then we have some preparation for the DNA replication. During DNA replication, it is a totally non-equilibrium process. The replication forks start replicating chromatin from A compartment to B compartment. And at the end, we end up with 2 polymer chains, and these 2 polymer chains, they condense further. They separate actually in G2M phase, in mitosis. Okay, and now there are 2 main, let's say, categories of chromatin modeling. The one is what is called top-down. These are mainly like machine learning-like approaches where we have like some interaction heat maps and we try to optimize them with some, we try to optimize the 3D structure with this data. Now most of these approaches are similar. In my opinion, they are quite limited, and it does not matter if like we have the highest correlation because a lot of the features in our data are just noise, and in my opinion, it doesn't matter. Bottom-up approaches are approaches actually that they are, let's say, based on physics. And in this situation, it is a bit, let's say, more closer like what we do, and what I will present you now, it is a bottom-up approach. But connecting with our previous discussions about, let's say, how machine learning connects with physics, I totally believe in a method that combines both of them to have both validation with experimental data, but keeping also the physical and biophysical knowledge. Otherwise, we will have to teach models like a lot of data that we already know from the physics that we know. Okay, now why we use stochastic simulations? Because I will speak about stochastic simulations. Actually, because we know that the structure of raw material is very dynamical, we have actually to solve an inverse problem of statistical physics. But usually when we speak about inverse problems in statistical physics, we mean that we know, let's say, some functional form of some Hamiltonian, and we try somehow to optimize it according to some data. In this situation, we don't know the Hamiltonian, so we have to make it up somehow. Yes, so I think this is the most important thing here. And now there are a lot of challenges. The one challenge is that there is not a consistent physical theory, so we have to do something for it. And I highly encourage you also to see problems regarding chromatin, because we don't have a lot of physicists that they work on that. The other thing, of course, is about the data, that they are very noisy. We have to find like the relevant features. We don't know what they are, what we have to keep from them. And the biggest problem that exists now is the validation, because if we don't have the experimental crystallographic structures, then we have no idea what is correct and what is not. But we have biological observations, we have data that they don't show the exact structure, but they show some features about regions that they interact together or compartments, and this is also quite important. Okay, so I start from loop extrusion. Loop extrusion is also a non-equilibrium process, but I think that we can approximate it as equilibrium. It is actually two players. We have two proteins. The one is SMC complexes. They are called loop extrusion factors, and they have this kind of ring topology. They bind on DNA and they extrude the loops. And the CTCF complexes, which are some elements that they say to SMC complexes that, hello, my friend, you stop here, especially when they have convergent orientation, because they have specific motifs that they bind. So we can define orientation of these motifs. Okay, there is also something else that it is quite interesting here. It is that when an SMC complex meets another one, then they stack together. There is not something like, I don't know, some particle behavior that they hit each other and they reflect or something. It is totally not like that. And in our data, we see some kind of loops in our experiments where we have like stronger interaction, we see a loop. So we have some interaction matrix from our data. These are called 3C type experiments. So I already explained what they are. The boundary elements and extrusion factors. We have this non-equilibrium process, and now we are trying to write down some energy. Yes. So before we write the energy, we need to write down the assumptions for our system. The one assumption is that if we have a DNA fiber, then its loop extrusion factor, it has 2 degrees of freedom, left and right. They connect two distance areas from DNA. The other thing that it is often, very often referred in the biological papers is that they cannot cross each other because they stack together, and the one SMC complex cannot pass the other. So we should somehow include this in our mathematical model. We have also this boundary activity of CTCF. There is also something very interesting that when once the SMC complex binds on the CTCF, then it starts to have longer lifetime because it remains there stable. And finally, that there is the tendency that loops extrude and they don't sink. Okay, so there are two ways of modeling. The one way is how all people do that, and the second one is how I do that. So the first way is by explicitly writing down all the rules and make some models of probabilities, things like that. So what I did, it was my first, let's say, paper. It is that I wrote down an equation, and this is, let's say, some kind of energy that I can maybe not minimize, but optimize on some Monte Carlo. And if I optimize this energy, then I can have, let's say, some possible configurations about how these loop extrusion factors can be. Okay, now the one thing is that I assume Boltzmann distribution. The microstates, which microstates here are the location of these loop extrusion factors, are following Boltzmann distribution. There is some temperature. The temperature is not actual, an actual biological temperature. It's just a parameter. Yeah. So the first term, it says pretty much that the loops have to extrude. The second term, it is a penalty for the crossing of these loop extrusion factors, and the third one is about the boundary elements. Is a data-driven term. It says that the energy becomes smaller when loop extrusion factor meets CTCF complex. Yeah, so we have some Monte Carlo loops, Monte Carlo moves. So we can have it either sliding in one dimension, because this is what loop extrusion factor do. And the second thing is that we can have some kind of mixing probability that one loop extrusion factor will rebind and it will bind in different places of DNA. We assume that we have a constant number of these proteins that they extrude the loops. And then we can calculate the energy differences just to have faster simulation and apply our metropolis or simulated annealing. And the reason why we do that, as I explained, is because we have non-equilibrium processes, and it is better always to have something stochastic that it does not give us only one possible, let's say, more probable or optimal state, but we can explore, let's say, near some optimal, some energy minimum. Yeah, it is better. Okay, so this was the first model. How I did that is first that we import some data about where are these CDC applications. Then we run this Monte Carlo, this metropolis, and we have some configurations of loop extrusion factors, and then we can very easily create some 3D structures by running some molecular dynamics, and then from these 3D structures we can have some distance heat maps, which we can compare their averages and also standard deviations with the data. And in this way we simulate loop extrusion. So loop extrusion is just one of the processes that we need to model the whole cell cycle. Now the thing is that most of the researchers, they model or loop extrusion, or there are very few models for replication, but there are not a lot of models that they try to combine the interplay of all of this. So here is what is DNA replication, is the process of copying the DNA. So for DNA replication, we have this kind of data, which are called replication timing curves. And what they show us is when whether a specific loci of DNA replicates early or late. As I said, A compartments tend to replicate early, B compartments tend to replicate late. Now, there is something very interesting that in general, to calculate compartmentalization in chromatin, we take high C matrix. High C matrix is some interaction matrix that we have from experiment, and we calculate eigenvector and eigenvalues. But in reality, when we have this data, we don't have to do something like that, because this replication timing profile, it is exactly the same as compartmentalization. It is like 95% correlated, except from some noisy features. Okay, so there is some discussion about how the replication origins start to fire. There was the assumption that they are dependent. It is not true, because we see that in a compartment they tend to replicate together. In my opinion, the main debate here is if it is only like one-dimensional thing, if the replication propagates only in one-dimensional or in 3 dimensions. Okay. And there are actually two kinds of quantities that we can calculate. The one is the replication fraction. This shows that if we have a population of cells, how many of them replicated, and how many do not replicate at a specific time and point. This is actually our replication timing curve. But what we need really to know to make a simulation is the initiation rate, which actually models the probability. And this is different, because not all the loci of DNA are potential origin. So some of them will have totally zero probability of replication. So while F replication fraction is continuous, the initiation rate is quite discontinuous. We need to separate which are the origins, which are not. Here I make a very big simplification. I assume some kind of Gaussian around the replication timing curve. This is a working assumption, because we want to integrate this process something a bit more complicated, and we have to simplify, as physicists like to do. So the process is very simple. The replication starts. There are some potential origins. We have some probability. We test if it will fire or not according to some pseudo random numbers. Once it fires, it propagates replication bidirectionally. And once all the DNA is replicated, the process has finished. Now, regarding the compartmentalization, this is something very interesting because it comes into the game the POTS teasing model. So we have A and B compartments in general, and we can assume that this is some kind of macro state of our system. So we observe this in a very, let's say, low resolution. And we assume that there is some kind of microstate that it is called epigenetic state, and it is responsible for that. The collective behavior of these microstates is resulting this. Now, I don't want to go very much into the biology of that. It's quite complicated. I think that also biologists, they do not fully understand it. And there are some papers that they try to model it with Potts model, but it is not so trivial, the connection with biology. But this is what I do as well. So the one way to go it is to assume that where is a loop, we have an interaction in the interaction matrix, and where it is not, the interaction matrix is 0. So in this way we can have some interactions, but these interactions change over time. So we don't have a static network. We have a network that changes over time. And of course we can have also an epigenetic field, which can be, for example, replication time of our data. This is modeled with block copolymer forces for, let's say, molecular dynamics. And in general, what we have at the very end, it is a coevolutionary graph. What it means that we have some network states. Here I have 5. Why? Because we observe now that, let's say, the most recent papers, they believe that there are 5 subcompartments. So it makes sense to have 5 epigenetic states. And we have also some edges, and these edges are actually our loop extrusion factors and the backbone. The backbone means pretty much that the loci I connects with I plus 1, just because DNA is linear, yes? So we have now a much more complex system. We have a system where both of nodes and edges evolve over time. And we can write down an energy for it. We take our previous energy for loop extrusion. We can add also Potts model, and we can also add some penalty about replication forks, because in general it is another one complication that once we have, let's say, the replication forks, then they also act as boundary factors for loop extrusion. And in general we end up with this framework. We have replication time data, then we make our stochastic simulation, we produce our trajectories of replication forks, and then we couple all the dynamics of loop extrusion into some kind of graph Monte Carlo optimization. And from these graphs we create a force field and we build the 3D structures. Now the thing is, okay, we have made something, let's say we have some complex simulation. The thing is why this is valid or not. So this is the most difficult part, actually, to convince biologists that this has some meaning, that it connects with their reality, how they know it. So before I go to that, in G2M phase, we have also some condensings. There are other loop extrusion factors which just extrude longer loops. And we have further compaction. This is also something, another one complexity that we have to model, because our simulation should have 3 phases: G1, S phase, where it is the DNA replication, and we have the potential for the energy for replication forks. And at the end, we have the compaction. We have the second population, which is faster, and topoisomerase activity, which means that we have some excluded volume and we disable it. Okay, so now because the time finishes, I want to show you some results. These are the 3D structures that we have. We can model indeed how the DNA replicates. I can do it for the whole chromosome. Now we can see some structural transitions between phases. In G1 phase, we see that we have some kind of equilibrium. There, our system at some point it becomes compacted, and then in S phase, because of the replication forks make the change to separate, we have decompaction, and then further compaction because of condensing the loop extruders that they propagate faster. So we did that. We also modeled replication stress. We observed that in replication stress we have shorter loops. For example, this is something that agrees with experimental data as well. This is some very new result because actually we have performed the experiments by ourselves in our lab, not me, but the biologist of our lab. So I repeated also for all chromosomes this analysis, and this is quite consistent that we have shortening of some loops, we have some pattern, let's say, in S phase. We made some validation, we compared with random models, we saw that our model reproduces better the chromosome structure. And also we have compared with single cell data, with high chip data that they tell us that some of the structural transitions are indeed valid, for example, the shortening of loops, or in S phase or in the replication stress conditions. We also observe that there is a very stable pattern of compartmentalization, and we give a kind of explanation for that mathematically, because in Potts model we can have phase transition when we have magnetic field only if we have a lot of long range loops that they dominate the system, and this is something that never can happen in chromatin because loops are in one order of magnitude smaller scale than compartments. So this is one possible explanation why this doesn't happen in reality. And we also compared with data with models, independent models from single cell data, and we also observed similar patterns. So in overall, we have a model which is maybe quite complicated, but it is not as complicated as we could think, because we use a minimal amount of data. We use actually only two types of data, replication timing curves and some kind of interactions about where are the CTCFs. And with that, we can model all the cell cycle of chromatin. Of course, there are some limitations about the parameter tuning. The reviewers asked me sometimes why it is better than other models, and I'm quite surprised because there are not other models. They model all the cell cycle. So, but it is still a valid question because when you make a model, you need to optimize somehow its parameters, and it is quite difficult when you have. So I want to finish. This is our team. I'm sorry I spoke too much, and that's it. Thank you.
Okay, thank you. So we have time for one or two quick questions.
Are you checking also a bit the transcription?
Yeah, the transcription, I have planned to model it, but for now, no. Transcription, actually, it is very similar to replication. We just need, like, it doesn't have a beginning and end. It happens totally random.
Yeah, I mean, with a model with enhancers and promoters, and it's well determined, no? For many genes, for many transcripts. The transcription, so the looping between the enhancers and the promoters. You will have like the high C. You will have
yes, I understand
matrix.
what you mean. If I see the looping between enhancer, maybe this is a good idea. Also, it would be a good idea that we would like to do in the future to make some maps of some genes about their distances, how it changes with replication and replication stress. But I haven't done it yet.
Okay, so let's thank the speaker again.
Thank you.
And all the speakers in this morning session. Okay, so we are gone. We have to be back here at 4 p.m., right? And a couple of messages before leaving. I think they are going to close this room, but they asked me to tell you that please do not leave valuable things here, just in case, so please take them with you. You have the link to the PDF in this QR code, just in case you didn't notice. And finally, in order to have lunch, there are many, you can go to the cafeteria in the Faculty of Sciences, and there are also many bars in Gonzalo Gallas, which is a street which is nearby. So just walk around a little bit and you'll find plenty of places to have lunch. Okay, see you later. Bye. Sí.